Good morning, everyone. Thank you once more for joining us after 2 long days for a third and final day on the meeting on the major transitions in evolution. It is with great pleasure that I present to you once more Professor Daniel Piñero from our Institute of Ecology.
Welcome everybody. To begin with the activities of today, we have of this excellent meeting, we have the pleasure and honor with the presence of Professor John Loxton from the University of Iowa, who will be talking about one of the mystery of mysteries in evolutionary biology, but not the one that Darwin called the mystery of mysteries, which was the diversification of the seed plants. I am referring to the origin and evolution of sex, and this theme mainly arises from the paradox that if sex through recombination increases, the variety of genotypic combinations of a highly adapted genotype will be diluted among the next generation. Most explanations given in the last part of the 20th century involve population processes and species interactions. Specifically, this includes first the adaptation under directional selection, that is adaptation to new environments through selection of novel gene combinations. Secondly, adaptations to varying environments, in particular, will evolve with adaptive genes combinations that are in linkage disequilibrium. And this will occur, for instance, within the context of coevolution with parasites and with rather strong selective regimes. This has gained considerable support since in some systems the existence of sexual reproduction is particularly common where the parasites are more abundant. Finally, another set of population explanation involves the separation of beneficial from harmful mutations through processes like Mueller's ratchet and the Hill-Robertson effect. These processes, using a population genetic approach based on mutation, drift, and recombination, would be responsible for the elimination of deleterious alleles. Professor Loxton's research goes further on these research lines, asking questions about the molecular mechanisms which underlie the molecular recombination process. To do that, he uses a comparative approach and species with diverse reproductive systems to disentangle, for example, the molecular mechanisms of parthenogenesis. This approach dwells on the enormous possibilities of proximal explanations in biology, how questions, to get a different look at proximate explanations, or why questions. Going back and forth from a particular species, from gene trees to gene function and vice versa, his research program has been an outstanding exploration and synthesis of new approaches to evolutionary biology. Professor Loxton did his BS in biology and psychology from Iowa State University, got his PhD in biology at Indiana University with Jeff Palmer, and did a postdoctoral studies at the Department of Biochemistry at Dalhousie University with Ford Doolittle. His first several publications started in 1999, addressed important issues like alternate hypotheses for intron origins, but also include several genomic papers addressing evolutionary questions in Archaea and Protoctista, as well as mechanisms of molecular evolution like gene duplication. Several of these publications appeared in Science, Nature, and the Proceedings of the National Academy of Sciences. I would like to stress one of the systems on which he and his research lab work. When one thinks about the origin and evolution of sex, rotifers come immediately to one's mind, in particular the existence of Deloitte rotifers, which have exclusively parthenogenic reproduction, suggests the use of these systems to study sex evolution. When I studied biology, rotifers included 3 classes. More recently, with the advent of molecular systematics, Phylum Acanthocephala has also been included within rotifer, otherwise a paraphyletic group. As a consequence, the study of closest relative of Deloitte rotifer would illuminate the origin of evolution of sex. This is the case of monogonont rotifers, cyclical parthenogens. Cyclical parthenogenetics appears to be a typical case of an evolutionary conundrum. While on the one hand it appears to be appealing, why is it not more common, only present in some lineages? Probably restriction related to cellular mechanisms limits its widespread evolution. At the same time, the existence of cyclical parthenogenesis in distant lineages suggests that this transition is not so improbable. Based on the idea that sex evolution should be closely related and explained by the evolution of the proteins associated with the meiotic processes, Professor Loxton detailed studies on these proteins have shed light on several aspects that appear relevant, like gene duplication, horizontal transmission, convergent evolution, and complex life cycles. Finally, I would like to stress another aspect of Professor Loxton's academic career. He is now the director of Pentacrest Museums, which includes collections related to the history of life, the history of Iowa, and the history of the University of Iowa. The Natural History Museum in particular was established in 1858. In Mexico in those days, we had the Reform War and the Reform laws, and we're beginning to delineate our nation for the 20th century, in particular the initial separation of the church from the state. But we didn't have any museum, natural history museum. This kind of cultural institutions should increase their importance in our modern society, and we should continually supporting them in any way you can. A relevant research system, the use of the most recent methodological approaches, clear-cut questions, and great evolutionary importance form the heart of Professor Loxton's research program. With that, I would like to acknowledge Professor Loxton's conference and leave the stage to him. Thank you very much.
Thank you for that very kind introduction. As it turns out, I'm no longer the director. I was the interim director at the Pentacrest Museums at University of Iowa for 3 years, and now I'm back in the lab, so I'm happy to be reinvigorating my science, and I'll tell you some about that today. But before I get started, I want to give special... Whoa! There we go. Excuse me. I want to give special thanks to the organizers, Juan, and my host, my handler, Deanna. And I also, I was very glad that Rick Michaud talked about John Maynard Smith yesterday, but more generally, I want to give deep thanks to Yosh as well about this book that this meeting obviously derives from. And I've used this book in multiple courses in my teaching. I've used it in my intellectual development, and when I first got the invitation to come to this conference and speak, I immediately accepted. I just checked my calendar, and then I immediately accepted, because this framework that they put together has had a huge impact on many of us, myself included. All right. So I'm going to acknowledge the people that did the work that I'm going to talk about. I'm going to start by acknowledging those folks. So during the phase while I've been museum director, my lab has reduced in size, but not in strength, I don't think. So this is my current lab as of last summer, really. And I'll point out work from people, some of whom aren't shown here anymore, that have moved on from my lab. So first of all, I have to thank Cindy Toll, my lab manager. She's been with me now 8 years. I call her my boss because she really does keep things going in my lab, especially while I was doing my museum gig. The work I'll tell you about a little bit is Elizabeth Savokal, who worked on the evolution of fungi, a recent PhD graduate. And then I'll talk about work on rotifers, as it turns out. I'll actually talk about some of the work on rotifers today, so thanks for introducing those interesting organisms that were done in collaboration with David Mark Welch and some former members of the lab, as well as some inventory work. The core of the work I'll talk about came from 2 previous graduate students, Banu Malik and Art Peitling. Okay, so I frame how I think about the origin and evolution of sex around this question of meiosis, because meiosis is, as I'll talk about in a minute, is sort of the core of sex. So here are the big questions that I see in this area. So first of all, when did meiosis arise? And this relates to these major transitions. So figuring out when it arose then allows us to ask then, how did it arise? In particular, from what prior functions did it derive? Because we know, and we heard from Simonetta yesterday and others, that that early derivation of eukaryotes from prokaryotes means that meiosis as a eukaryotic specific trait had to have evolved from some other pre-existing function. Given that it did arise, how has it subsequently evolved? And in particular, I'm going to try to get to this question, although I probably won't answer it. Can meiosis be lost? And this is where deltoid rotifers and other organisms come in. And more generally, and I think this gets to, and in the introduction it was mentioned that sex, the evolution of sex is a sort of outstanding issue in evolutionary biology, and it's still very active. This sort of much deeper question, a sort of career, maybe multi-career, multi-generational question. What are the consequences of meiotic evolution? This is what I'm doing. But for this more general concept of the maintenance of sex, why do we do it? Why do organisms do it? Okay. So I'm going to start by generally talking about the theories for maintenance of sex, for the persistence of sex, really. And I call them the good, the bad, and the ugly. And so the good, and these are not mutually exclusive, okay? But these are sort of generally the sort of three ways that scientists have thought about why sex has been maintained. The good is increased variation is beneficial to adaptation, and this is attributed to Weissman, and of course extended by Fisher. Sex brings all the good mutations together. Okay, so there they are, and the bad. Okay, deleterious mutations will accumulate in asexuals. So there's the flip side, and of course attributed to Hermann Müller. And the ugly, I call the ugly. And this is a kind of more recent set of thinking, which is that sex allows adaptation to changing environment, and including this idea of the Red Queen, and in the special case that parasite genome coevolution. Okay, so and this is attributed, of course, to Bill Hamilton. Okay, so there's and as I said, for any given system, some or either one or more of these things actually might be responsible for the maintenance of sex, and there's been a lot of work trying to look at which one is more important. And my thought is that probably one or more is important in most organisms, but the strength of the effect of these various reasons are going to be different in different lineages. Okay, so here's the core of my talk today. I'm going to talk a little bit about eukaryotic phylogeny, and this gets to this question of when did sex evolve, when did meiosis, what is the phylogenetic distribution of meiosis? And I'm glad to have been able to switch my talk to today, because you've already been informed about eukaryotic phylogeny and deep and LICA and that sort of thing, so there's a lot of work that I don't have to do today. And the work that's been done in my lab over the last 1.5 decades, I guess, are developing these molecular markers for sex, which are meiosis-specific genes, and their phylogenetic analysis. And so I'll tell you about a lot of different genes. Here are a couple of them: the RECA homologs, RAD51, and the meiosis-specific DMC1. And then there's a whole bunch of other genes and many, many more species. And what we do with those is we take this sort of phylogenomic inventory of the meiotic machinery. So, and particular organisms have played important roles in this process. These putatively early diverging organisms, which I'll talk about to some at some length Giardia and Trichomonas as exemplar, potentially early diverging protists, and allows us to ask these kind of questions. What components of meiosis are present among diverse eukaryotes? And where did meiosis genes come from, and subsequently how has meiosis evolved? Okay. And then finally, I'll get to the end and talk about deploying this kind of historical framework, this inventory, if you will, to detect hidden sex, perhaps, or the lack thereof. So I'll talk about our work on deloid rotifers as a test case of putative ancient asexuals to get at this question. Is it possible, is meiosis so important for being a eukaryote that it can't be lost in the long term? Okay. All right. So I'll start with the yin and yang of sex. And what I'm going to talk about, usually when we talk about sex, amongst company, we'll usually be talking about the sort of getting together of gametes, right? That's sex, is the getting together of gametes, or the processes that eventually lead to the getting together of gametes. But that's only one side of the equation, because of course the other side of the equation is the generation of gametes, which involves this important biological phenomenon called meiosis, which reduces diploid cells to haploid cells that then are part of that process. So when I wanted to study the evolution of sex, and I did want to study the evolution of sex, I thought about, well, as a molecular biologist, of course I want to study some genes. And it turns out that many of the genes that are involved in this sort of getting together of gametes, okay, fertilization, et cetera, the fusion of gametes, it turns out that many of those genes are actually very fast evolving. Okay? In fact, some of those genes are actually among some of the fastest evolving genes that we know about, right? But it turns out that many of the genes involved in this process, meiosis, are actually reasonably slow evolving. They're highly conserved, and I'll show you some information on that. But in any case, so to study this process over the 1.62 billion years, depending on who you ask, history of eukaryotic evolution, one needs to actually have genes that are reasonably well conserved. Okay, so probably don't need to tell you all, but sex is the predominant reproductive mode amongst eukaryotes. Most organisms do it at least as part of their life cycle, not necessarily all the time. So pretty much everybody's doing it amongst eukaryotes. Asexual reproduction, however, is actually fairly widespread amongst, again, a diversity of organisms. There are deloid rotifers right here. But generally not deemed to be very successful long-term. In other words, there are not very many, if any, lineages that have persisted without sex for a very, very long time. And so asexual reproduction seemingly has short-term advantages which don't pan out in the long term. Okay. So here's a little bit more about ancient asexual lineages. So they persist long. So this is a review that we wrote a number of years ago, sort of documenting the organisms that seem, from the literature, that seem to be persisting longer than one would expect, on the order of millions of years. And so I'll just, this is just a rogues gallery of these organisms. Things like, these ambistomas, orbated mites, arbuscular fungi, various kinds of ferns, and something I'll tell you a little bit about is this organism, Meet Giardia. Hope you haven't met him before. Anyway, these are all organisms that have thought to have not had sex lives for a long time. And so these would be particularly important to investigate if sexual reproduction is so important, then figuring out how these organisms get away without doing it would tell us something about why sex is important. That's the main shtick of this kind of angle of work. Okay, so who is doing it? So I put together this slide quite a long time ago, but I still like to use it because it emphasizes a couple of things. So let's look over here on the right side. So here's a phylogenetic tree of eukaryotes that's quite cynical, actually. But it basically points out that the phylogenetic part is showing a huge lack of resolution amongst the eukaryotes, which has been improving, and I'll say some things about that. But in any case, what we know about sex across the tree of eukaryotic life is actually pretty minimal. Of course, most of the organisms on the eukaryotic tree of life are unicellular eukaryotes, and of course not that many people study them, so we actually don't know that much about their sex lives. We know that, of course, most, if not all animals are sexual, fungi, et cetera, et cetera, et cetera. So we really don't know a lot, and there are some organisms that had been studied enough that people had made the ascertainment that they probably weren't having sex. So things like Giardia and Trichomonas. So this is kind of out of date, but it sort of points out sort of the state of knowledge when I first started thinking about this problem. So where do we get evidence for sex or lack thereof? So evidence for sex traditionally, before the work that I'm going to tell you about, came from either population genetics or from direct observation. So people looking for recombination in populations, or by catching them in the act. Right? However, the problem with this, of course, is that absence of evidence is not evidence of absence, and so does that mean just because we haven't seen them doing it, does that mean they're asexual, or are they doing sex on a sort of covert or infrequent way? So indeed, even population genetics approaches sometimes cannot reveal very infrequent sexual reproduction. Okay, so I wanted to think about some way of looking at this question without at least these issues being so important. The other thing that sort of framed up how to think about this problem that was important to me. So this is the phylogenetic tree of life when I was a graduate student. This was the phylogenetic tree of life, and it was really inspiring, of course, that we could do this and we could place all the organisms in one framework and set us up for hypotheses about how things have evolved. A couple of things to point out, of course, is that we have the two groups of prokaryotes and we have eukaryotes here, and a couple of things to point out is, and this is, of course, based on small subunit ribosomal RNA. And based on this, we sort of had this view that animals, plants, and fungi were very late inventions in the eukaryotic tree of life, and that there was a sort of ladder-like evolutionary history of eukaryotic protists. In particular, what really got a number of us going in these years were these organisms here: diplomonads, trichomonads, microsporidia. As notice, they're the first branches in the tree of eukaryotic life, and it's not a spoiler to say that it turns out that's not to be true, but this is where we were starting, and this Archezoa hypothesis, which was discussed yesterday, I think, was based on this idea. And so there are some pictures of those three organisms, and it turns out what was really important to me was that all three of those lineages were not known to do meiosis. They were asexual. They were not known to do meiosis. They also didn't have mitochondrion, which subsequently found out that they actually probably had one and lost it. So this is sort of what we call the meiosis late hypothesis, is that meiosis evolved sometime during eukaryotic evolution, such that that these represent sort of the ancestral state of eukaryotic cells that didn't know how to do it using meiosis anyway. So that was, and this gets to Ignacy's point, which is that you can have this sort of yesterday about the evolution of animals, which is that you have this sort of nice starting point where you have a group that doesn't and a group that does, or whatever thing that you're interested in. So this is perfect for these sort of major transition kind of argument, which is that I could look at the genomes of these organisms, figure out what they did and didn't have, and then look at the ones that evolved later and see what they had, and then wow, you can figure it out, right? So that was where I started. I'm going to tell you that turns out not to be true, but it turns out that so now this is you've seen various iterations of this, and it's changed a little bit, but not actually very much. So this is now moving forward a number of years now, we're seeing that a couple of things. Well, so let me just say. So we had this ladder-like view of protist evolution, but we move forward in time and we actually lose resolution among the major eukaryotic groups, such that and we have some resolution now, but in any case, in this kind of view that has persisted for a number of years, and in fact, we still don't know what the root of eukaryotic tree is. It's been being debated in literature as we speak. That it might be that these organisms don't represent the earliest branch on the eukaryotic tree of life, right? So that, however, at this point, we now know that there's a lot of information about most of these eukaryotic groups, most of which contain protists, actually all of which contain some protozoa. And it led Alistair Simpson and Andrew Roger, my colleagues, to write this paper where they say the last common ancestor of living eukaryotes now appears to have been a complete eukaryotic cell. It had a nucleus, ER Golgi. Simonetta showed a slide that had some other things on it, actually, that we've learned subsequent to this. Underwent mitosis and meiosis. And that turns out probably to be true. We didn't, and what I'm going to tell you is that this was a little premature for them to say, but I'm going to tell you that they were actually right. I'm going to show you the data that indicates that, in fact, they were right. And that leads to the alternative view, which is the sort of, one might call meiosis early as opposed to meiosis late. So now getting back to this argument about the before and after, that means that if meiosis is early, then that means that the closest comparator to a cell that didn't have meiosis is a prokaryote, which is kind of too bad in a way, but that's the way biology works. Okay, so I'm going to tell you about that today. All right, so here's back just for a moment, just back to the ribosomal RNA tree of life, and you're here with your fungal colleagues, and I just wanted to. The main point of this is to show again, sort of. Is eukaryotic diversity, regardless of what the details of this tree are, is that eukaryotic diversity is largely represented not by animals, plants, and fungi, but all these other various protist groups. But unfortunately, and fortunately for me actually, but kind of unfortunately, is that we really only know about how meiosis works from a very, very limited set of organisms. And so now I've taken animals and fungi and blown them up onto this whole slide, with plants here as just a little outgroup. So I put this slide together a number of years ago for a meiosis Gordon conference that I attended, and I put little boxes around organisms that were being discussed at that meeting, and you can see that almost all the organisms that were being discussed were animals, fungi, and there were a few plants. Okay, so what we really wanted to do was to take the information about how meiosis works and the genes, et cetera, in animals, plants, and fungi, and then use that information to go looking for those genes in more diverse eukaryotes. So we went about trying to do that, and it's kind of hard to imagine now how fast genome sequences have influenced our way of thinking. So as of about 2005, so now here's this eukaryotic tree of life with the sort of major supergroups, if you will. Root positions unknown, et cetera. And I have boxes around groups which contain at least one, genome available for analysis. And you can see that some of the big groups in these unplaced things don't have genomes at all. Some of them might only have one genome, et cetera. So we had very sparse coverage of genomes across the eukaryotic tree of life. And here again, what we're talking about is that if I'm interested in studying meiosis and I'm finding genes that are important in animals and fungi and to some extent plants, and I want to go see if they're present in more diverse eukaryotes, you can see how sparse that kind of analysis is going to be initially. So now fast forward now to a few years ago. Now this is 2011, and it turns out we now essentially have coverage of at least one genome, and in many cases multiple genomes from all of the major eukaryotic supergroups. Okay, so that's been a great movement forward in terms of data available to answer questions. Okay, so that's the setup for the sort of phylogenetic part. Let's talk a little bit about meiosis. So this is my University of Iowa meiosis slide. And meiosis is obviously this sort of central process responsible for sex. And just a reminder, is that all the good stuff happens in meiosis 1. Okay, so recombination, et cetera, et cetera, et cetera. And meiosis 2 happens, which uses much of the same machinery as mitosis. So really the genes that we're interested in are occurring here. And at each of these various steps in meiosis, there are many proteins involved in each step, and several of them are meiosis specific. Okay, a little bit more about meiosis 101. So here's mitosis by comparison, and so all of the important things that are going on in meiosis 1, pairing, recombination, the segregation of centromeres in a way that's such that for the second division, then you maintain the centromere such that you can do a mitotic-like division in the second division. Okay, so it's all of the special molecular machinery going on here, much of which is actually concentrated on this generating crossovers, synaptic complex, et cetera, that we can study their evolutionary history. A little bit more about the details. So this is now the double-strand break repair model for meiotic recombination, and the meiosis-specific genes are shown in green. I don't know whether you can see them, but there are lots of genes involved, and this is just a subset of the genes, of course. Is it you get cohesins binding, you get double-strand break formation. This is an important gene, SPO11, which actually makes double-strand breaks in DNA, which would generally be a bad thing, but it's important for generating crossovers, binding of recombination proteins, et cetera. So there's a whole series of steps that happens, all of which include at least some meiosis-specific components. Now, the other components, the ones in black, are actually not meiosis-specific, but they're involved not only in meiosis, and many of them are involved in either DNA repair or mitosis. So they have sort of dual roles in the cell, but they're deployed during meiosis to collaborate, if you will, with these meiosis-specific versions. Okay? The other interesting part of it, so how can we use meiotic genes to study the evolution of meiosis? So I had to set up some criteria. I've already mentioned this idea of them being meiosis specific. Now this is a little bit of a tricky one. For a gene to be meiosis specific, they have to be expressed and or required only in meiosis. And given the number of model systems that we have available, we have fairly limited information, but consistent information. So, for example, many of these genes that I call meiosis-specific are known to be meiosis-specific in multiple animals and/or multiple fungi. We don't actually really know in these other organisms, but the inference is that they're meiosis-specific. They're conserved enough to study. I've mentioned this already. Many of them actually have prokaryotic outgroups. They actually have homologs that are in prokaryotes that are not doing meiosis, obviously, that are doing something else. Turns out that eukaryotic gene duplications are really helpful, and I'll talk a little bit about that in a minute. So, when I started this sort of research program, it's too bad that I just couldn't, be God and decide which, that these were going to be the best way to do this. But anyway, if I were, I'd say the most useful genes would be present in everything that was known to undergo meiosis. So if, all the organisms that are doing it, they would have all of the genes. And if the organisms weren't doing it, they would be absent, right, under a use it or lose it kind of model. Okay. Turns out biology and evolutionary biology are as messy. Those things don't work 100%, but that's sort of the general idea. Okay. This is the other thing I wanted to mention is that the evolutionary history, the molecular evolutionary history of meiosis is already indicated by just simply a description of how meiosis works. So this is the double-strand break repair model of meiotic recombination. It's an old paper, but it makes the point, which is there are a bunch of, and these are yeast gene names. That's been another fun part about studying meiosis across lots of organisms, is that everybody names their genes different things. So, but anyway. So here are the yeast names. And so there's a bunch of genes that I mentioned that are involved in both meiosis and mitosis. These are obviously not meiosis specific. And then there are ones that are involved in meiosis only. So these are the meiosis specific ones, okay, at these various steps. But what the boxes are they so the boxes represent genes that have evolved by a gene duplication. and the ones that have colored boxes are ones that are paralogs of each other. So I'll talk a lot about these today. So RAD51 and DMC1, these are actually versions of each other. DMC1 is a meiosis-specific one. These are mut-S homologs. They have meiosis-specific gene duplications, et cetera. So the ones that are in boxes that aren't colored have gene duplications that are doing something other than a double-strand break repair. Okay. So we already know, just based on this alone, that many meiosis-specific genes have evolved by gene duplication from genes encoding more general functions. So that tells us already something about the origin of meiosis, is that gene duplication had a very big role in generating this sort of novelty of function. All right, so I'm going to walk you through what might be kind of eye charts. But this is the kind of way that we've been doing this for a number of years, and they're complex, but they actually are in some ways sort of simple representation of this so-called inventory of meiotic genes. And I'm going to have to walk you through sort of a number of iterations of this that have gone on in my lab. So this is one of the early ones in 2002, and I've got the protists on top. I don't have humans on top. So I have protists on top, and I have animals, fungi, plants, et cetera. So these are representatives of genomes that were available to me. And I've noted down here homologs that are present in either archaebacteria or bacteria. Okay? So the yellow columns are genes that are meiosis-specific. The white columns are these more general functions. So you might say, well, why am I caring about the white, the ones that are not meiosis-specific? They do two things. They tell us something about our sampling, but they also provide a context for the history of these meiosis-specific genes, many of which are paralogs of each other. Okay, so back 2002. First of all, I'll point out that SPO11. So this is a gene, the meiosis-specific gene that creates those double-strand breaks, okay, that are required for recombination. It's a really interesting gene in that it's hard to imagine. It's hard to imagine a gene that actually has another function in creating double-strand breaks, except for as it is being used in meiosis. Now, it turns out its homolog in archaebacteria is actually a topoisomerase, which also creates double-strand breaks. But eukaryotes don't use this topoisomerase. Okay, so this is very specific to eukaryotes, and it creates double-strand breaks. So generally, you don't want to be creating double-strand breaks unless you're pretty sure you're doing meiosis, right? You're going to use those in an effective way. So at this time, SPO11, everybody's got SPO11. It turns out, and you can see that most of the columns contain pluses. Each of these pluses and minuses are ascertained. The pluses are actually ascertained very rigorously by phylogenetic analyses. The minuses are always, again, it's part of that absence of evidence. Although most of these are actually high-quality genomes. One of the things I'll point out is that early on we started seeing organisms that we know have sex, like Drosophila and C. elegans. We're pretty sure they have sex. Yet they're missing things like these genes here, right? So they're missing, and they're missing whole swaths of them. That's really interesting. So, and we're pretty sure they're missing them. And so it turns out that we have cases where there have been independent losses, okay? And why I have bombyx in here is to point out that actually they were independently lost in both flies and in worms, okay? So, and it turns out that almost the same set were lost in a group of fungi. And I'll get back to this group of genes in a minute. So we started, again, it was starting to violate this sort of everybody's got them or everybody doesn't kind of idea, but it poses really interesting questions. Turns out that those genes, at least HOP2, MND1, and DMC1 work in a protein complex. You lose one, you lose them all. So we've learned something about how meiosis works from these kind of distributions. That kind of approach led us to go straight after the jugular, really, which I mentioned early on, which are these so-called early branching eukaryotes. And again, sort of influenced by this archezoa hypothesis that Giardia and tricho and these various other organisms might be the earliest branches, and they weren't known to have sex, so yeah, they aren't going to have the stuff. Well, the Giardia genome was being completed, and at the same time I was doing lots of PCR. But in any case, we published this paper back in '05 in which we surveyed through a variety of these genes, and in this case, the meiosis-specific ones are in gray. And Giardia had most of the genes, including most of the meiosis-specific genes. They didn't have this set, MSH4 and MSH5. That was interesting, but for the most part, they had the stuff. So this led us to sort of two thoughts. One is that it was providing evidence for sex. So they had a number of these genes that were meiosis-specific in anything that had been studied sufficiently to determine their meiosis specificity. And if Giardia represented either the earliest or among the earliest branches, it would suggest that there would be an early origin for meiosis, right? So they've got those two things kind of combined, okay? So it led us to those two kinds of thoughts. As it turns out, remember I was saying that one way you could look at the presence, looking for sex in organisms is to go do population genetics. A few years ago, it was very satisfying to find out that a group of researchers went out and actually did population genetics in Giardia and found that, in fact, they actually have recombination that is diagnostic of meiotic recombination. Led me to, I got to write this nice little piece. I don't know if it was nice, but it was fun to write about it. So we sort of tied two things together. We took the meiotic inventory, which suggested, yeah, they probably are having sex. These people went out and actually used this other method and said, yeah, if we look hard enough, we're actually going to find evidence. Later on in work that I'm not going to tell you about, it turns out they actually have a very strange kind, what I think is actually a meiotic division called diplomyxis that Zach Candy and his colleagues have discovered. So it probably has come full circle in Giardia. If you look hard enough, you have enough clues, you can find evidence for meiotic process in these organisms. And to be honest, the diplomyxis that they found in Giardia was actually quite strange. And in fact, some people might argue it isn't meiosis, but certainly it's a derivative kind of reductional division, probably. Okay, so now I'm going to move forward a little bit to a little bit more recent surveys. So we had Giardia, another excavate, Trichomonas, which was also not known to have sex. And so what was interesting is that Giardia, and here's Giardia up here. Now the colored boxes are shown here, and if they're present, and then et cetera. So one of the interesting things that you'll notice is that when shown this way, you can see that yeah, there's a lot of organisms that are here and there missing various genes, right? So assuming that we've ascertained their absence, which isn't entirely clear, but for the most part, I think these absences are relatively clear. It suggests that some of these genes are actually being lost occasionally during evolution. But what was interesting from Trichomonas is that it had not only in the number of genes that we surveyed in this paper, I think it was 27 if I remember right, and Giardia is missing a whole bunch of them. Tricho is only missing 2 of them, and it was among these earliest lineages. And compare that to us and yeast and Aspergillus, which have the whole set. And so you can already see that these genes are already in place, at least at the divergence of Trichomonas and us, which is actually might be the biggest divergence among eukaryotes. So Giardia is missing more than Trichomonas. So that means, but it has most of the stuff. So Trichomonas is probably doing it too. But we wanted to compare the meiotic machinery in Giardia. So the meiosis-specific ones are in red here now, and in Trichomonas. And remember, I pointed out that MSH4 and 5 were missing in Giardia. Trichomonas had them. So when we first saw this result in Giardia, we thought, oops, this was the tree we published with the Giardia. Here is the phylogenetic tree of MSH4 and 5, and the branch colors are the groups of organisms. So brown is fungi, red is animals, green is plants. And when we published this, we saw, wow, MSH4 and 5 as meiosis-specific paralogs actually don't have any protists. So maybe, even though maybe meiosis might have evolved early, maybe this particular aspect of meiosis evolved late. Turns out not to be true either. So as I already told you, Trichomonas has them, and Banu, my student, went out and looked more carefully amongst a variety of other protist genomes, and the protists are kind of in this sort of orange color now, and she found, basically protists have these too. And so it was an ascertainment problem, right? So we just didn't have enough genomes at the time to know this question. So MSH4 and 5 also present at least as early as the rest of them. That's a kind of object lesson in finding data. So this, I think, is the last eye chart. Now you know why I call it an eye chart. Showing sort of, what kind of distribution we were working on, so lots more organisms, lots more genes, et cetera, and so this is sort of the end of Banu's PhD thesis, and so you can see that the distribution of these genes is actually quite broad, and these dots down here at the bottom represent those ancestral states in prokaryotes that were that homologs of these genes were already present. Okay. I forgot I had this in here, but this is fun to say. So we're talking about MSH4 and 5. So MSH4 and 5 are actually involved in what we call crossover resolution and crossover control in the meiosis field. And it turns out that the MSH4 and 5 have been lost many times, but it turns out, where'd it go? Crap. It turns out that that there's another pathway that was discovered maybe about a decade ago that have this sort of other pathway that occurs in parallel with this sort of crossover resolution pathway that's encoded by MSH4 and 5, and it's this MOS81 MMS4 pathway. And what was really interesting is that in the organisms that didn't have MSH4 and 5, they had the other pathway. Some organisms had only the MSH4-5 pathway but didn't have the MMS pathway, and some organisms had both. So it's one of those situations where when you survey through these genes, you can see patterns that tell you something about not only their evolutionary history but their functional roles. Okay, so I'm going to bring you kind of up to date. Art Peitling sort of decided to sort of take this one step further, and at this point now we're coalescing on a eukaryotic tree of life that has these sort of major supergroups. The names are being changed as we speak, I think, but in any case, they sort of coalescing into these supergroups, which I have in different colors. So here's animals and fungi and various protist groups. And so the colors will kind of remind you of sort of what the major groups are. You don't even have to know what they are. And so here are the genes. Here's a survey of a select set of organisms. And when you do this kind of result, you can then say, okay, we can now look by group. We can say, okay, can we figure out what the common ancestor of that group would have had by inference of the members, the presence or absence in the various members. We've seen that kind of logic a few times in this meeting already. So when we do that, we sort of sum, if you will, by group. This is the result. And so here are the genes, here are the groups. And so what you can see is that some of these genes are actually somewhat sparsely distributed among some of the groups, so they're either not present or not, or we can't find them, right? But most of them are actually present in at least one member of that group. So if they're present in one of that member, that means that they're present in the common ancestor, right? Unless lateral transfer is going on, but I'm not going to get into that, and I don't think it's happening, to be honest. Okay, so if we do that, then we can say at least 10 of the meiotic genes. Now, these are just not the meiosis-specific ones, so this is our 20 set. Were present in the last common ancestor to all eukaryotes, assuming that we have now coverage of all of the major groups of eukaryotes, that we don't have any leftovers. Okay? which I'm reasonably confident of, but not totally confident of, right? So, but in any case, so given that we haven't found any new major eukaryotic groups, this would then, so that would mean that these ones are certainly present in LICA, right? So this one might maybe not. If amoebozoa were the deepest branch, then then it might not have been LICA, right? But only if that one was the deepest branch. Is everybody following this logic? Okay. And Art actually developed a metric for missing data as well, which I won't tell you about. But it's actually kind of interesting that we have more or less confidence in missing data depending on the quality of the genome. Okay. However, we have this trick, and the trick is really super important. Turns out that I already told you that many of these genes have evolved by gene duplication. So here's the trick. If they have evolved by gene duplication, so you have one paralog in the other, then the presence, assuming that they evolved by a gene duplication before the origin of eukaryotes, right, which I'm going to show you, then the presence of one or the other paralog means that they had both paralogs. Okay, I'll show you that. So that means that even though an organism might be, in species 4, there might be missing this particular paralog, if they have this paralog, then it means that they had it and lost it. Okay? Does everybody follow that logic? Because this gene duplication predates the diversification of eukaryotes. Okay? So if we do that trick, then all 20 are actually present. So we now have summed across all of the organisms, but we've also then combined the ones that are duplications of each other. So either one or the other specifies that gene was ancestrally present. It may have been lost, or we maybe can't find it, but it had to have been present. Okay, because the importance is that those gene duplications predate the origin of eukaryotes. I'm going to show you that in a minute. Okay, so here's the current meiotic gene inventory. And we can do these. We can actually analyze the data and construct phylogenetic trees. We've sort of gotten out of the eukaryotic phylogeny business, but it turns out that these genes are just as good as making trees as any others, and they reconstruct a reasonable tree. So here's the summary. If we basically collapse them into the major groups, and here are the gene duplicates. So here's RAD21, and here's REC8. These are the ones that have evolved by gene duplication. We can make that very strong inference about their presence. So here are the phylogenetic trees that show, and it turns out that these meiosis-specific genes have evolved by gene duplication, and they aren't just all in one sort of functional category. So we have ones that are involved in synaptonemal complex formation, axial element, cohesins, double-strand break repair, crossovers, homologous recombination, et cetera. So all the major sort of functional processes in meiosis, the core genes involved in this process have actually evolved by gene duplication. Okay, so and some of them actually have evolved from genes that I haven't talked about. So like HOP1 has evolved by gene duplication from this gene called REV7, et cetera. And all the gene duplications happen. In this case, so here's an example I can show you. Here's RAD51 and DMC1. That's the archaebacterial homolog. Okay. All right. So really what that means is that meiosis itself, that process, that novel process, this major transition, if you will, had its sort of origins in a massive amount of gene duplication that then gave rise to new functions that then gave rise to this process that we call meiosis, right? Related to mitosis, but not specifically. Okay? The details of which now, since I don't have these intermediate steps, that we have to look to archaebacteria, and so there's a big gap between prokaryotes and eukaryotes. So on the good side is we can say, okay, yes, meiosis arose early by gene duplication. LICA had meiosis, but it's hard to understand that transition because we don't have intermediate steps. And that's the sort of too bad part about meiosis late, because had we had meiosis late, that might have worked, but biology is too interesting for that. Okay. All right. All right, so I'm going to tell. So given that now, my research program kind of started changing because I no longer could study as much the origin of meiosis. I had to study its evolution because I was still interested very much in meiosis and how it's evolved. So that led us to start thinking about, well, given that it's there, how has it evolved? An interesting question, maybe not equally interesting. It's up to you. But in any case, so we started studying the evolution of sex and meiosis in particular eukaryotic groups, and we started working on this on fungi. So here's a diversity of fungi. Here's a phylogenetic tree of fungi. Turns out we started working on this because there were a whole bunch of genomes being done at the time. We knew a lot about meiosis in some, and it turns out there were lots of weird sort of processes going on. How am I doing on time? Oh, how much do I have left? Oh, okay. Anyway, it's 10? Okay. So I just want to tell you a little bit about work that we did in fungi, and it turns out what was really interesting was that we had very detailed and broad phylogenetic sampling in fungi. And so this is the work of Elizabeth Savocal. And here's this case that I was telling you about earlier, is that we started seeing patterns, like, so if you're missing one of these genes, you were missing all of them, et cetera, and this would happen independently. So this tells us something about how meiosis evolves. This is Elizabeth's PhD thesis, and it's sort of, she did, 100 genomes so 100 genomes by 15 genes, and you can see that most of the fungi have most of the genes, but you can see these nice patterns, right? So this tells us something about how meiosis evolves, and what's interesting, what she actually found that required a sort of detail was that she found cases of genes in the process of being lost, and not, and it turns out that there are genes that I've already made you familiar with, which are MSH4 and 5. So in these organisms, MSH4 and 5 are actually pseudogenes, and the only reason why we could find them was because we knew their synteny, we could find the flanking genes, and we could actually find them in the process of degrading. So that was sort of in our use it or lose it kind of model, but these are sexual organisms, so they presumably are losing this particular set of genes. Okay, so there are some interesting things that came out of that. And you can see that in many cases that genes have been lost independently. So here are the highlights. We found cases of these sort of groups of related functions becoming pseudogenes. Oh, it turns out that I didn't tell you about ZIP3, but it turns out this protein also works in the complex with MSH4 and 5, and it's also being lost. And not only lost because we can't find it, we can actually positively find it as a pseudogene. So that's not the absence of evidence, that's true evidence, right? So that tells us about how meiosis evolves. We have this weird case in this organism, Malassezia. It's every time I give this talk, I scratch my head. It's the dandruff fungus, and it turns out it has almost none of the meiosis-specific genes. But so we don't really know anything about how meiosis works or doesn't in this organism. So this is a super candidate for investigating further, as a kind of exception to the rule. Okay. So we learned from this kind of thing, and this is a super summary from a very detailed thesis, is that we could learn about loss, which genes get lost. Some of them we learn about the pathway of being lost, the order of being lost, et cetera. So this tells us something about how meiosis evolves, is that genes are being lost, and presumably these functions are at least somewhat dispensable, maybe being replaced by others, or maybe there are backup functions that are taking place. Okay, so this is a summary of this part. There's your eukaryotic tree of life, and there are two major points, which is that the molecular machinery for meiosis evolved once and early, prior to extant eukaryotes and LICA. That's it, right there. Right? There's no evidence of any organism that diverged earlier than that. That doesn't have meiosis. And if there are any putative asexuals, they are derived from sexual ancestors. So I've got a few of them sort of marked here that were putative asexuals, and they're all derived from sexual ancestors. So sex came early, and if it's been lost secondarily. That probably sounds familiar to things like mitochondria and things like that. Okay. But what about everybody else? And this is sort of the fun part. You know, we only had a small number of genomes available to this kind of way of thinking. We have a whole eukaryotic tree of life to look at. So we wanted to sort of take this approach as what we call a meiosis detection toolkit. There are a lot of these organisms that are not known to have sex. We already know, and I showed you lots of data that the presence or absence of one gene is not enough because these genes, are being differentially lost. But if we had a set of genes that could be isolated from a broad diversity of eukaryotes, this might be useful. So in other words, looking for the genes in organisms as an ascertainment about whether those organisms are capable of sex. In the end, we want to know, is everybody doing it? And the bigger question is, that I've already alluded to, is meiotic sex required of being a eukaryote in the long term? So is it really truly possible to lose meiosis? Okay, so here's my general setup here. So we want this to be general, so we want to be able to go to any eukaryotic organism and ask this question. So I went out to the literature and I found a good example. You don't want to know what my Google cache looked like after I did the searches to find an example for this. But anyway, I found this example. SpongeBob, as it turns out, according to his creator, is asexual. Why he's called Bob, I don't know. Because if he truly is asexual, he ought to be Spudge Barb, right? Anyway, so according to his creator, SpongeBob is an asexual. So we're gonna actually. This is now going to be a research grant in my lab. I'm going to figure out whether SpongeBob is actually asexual. Here's what we're going to do, obviously. Not going to do that to start with. That's what people would have done. But we're going to do this, right? We're going to say, are the meiosis genes present? If so, I might do what they did in Giardia and call up a colleague and say, okay, yeah, can you study population genetics of SpongeBob and tell me whether he's having recombination? That might be worthwhile doing. I'm not going to do it. Okay, so how many? Which ones? If he has one or two, or if it's limited to a few, maybe SpongeBob is asexual. Are they expressed? Are they, and in particular, are they? Is there something about their developmental, temporal, and spatial pattern? If they're all expressed, say, right here, at the same time, in the right order, then SpongeBob's germline's up here. Unlikely to be down here, probably. But anyway, so when and where? Because we actually know something about how those genes are deployed. One possibility is, and Iñaki, I'm super jealous of being able to take genes from one organism and put them in another organism to see whether they work. We've not gotten this to work, but it's an idea, right? So you potentially could take SpongeBob's sex genes and drive sex in yeast, let's say. Hasn't really worked very well, but one could do that. In other words, can we induce meiosis? Because we're not going to do all the work to figure out how SpongeBob has sex until we have some clue that he might be having sex, right? So can meiosis be induced? If not, he's not a putative asexual, and we don't want to know. We don't want to know what's going on with SpongeBob. Okay. So that's the general setup, right? So we would want to have a general way of looking at this question, and then going in and looking. So I'm going to tell you about rotifers. So these are the SpongeBobs really that I'm going to tell you about for the rest of the talk. As was already discussed, these are deltoid rotifers. These are putative asexuals. They're all females. These are monogonate rotifers. They're cyclical parthenogens. They alternate between sex and asex. Sister group to these. Okay. So this is a before and after. This is a good one. Okay. The phylogenetic relationships shown here. They're Alophotorozoan. And they fall into these groups, and it turns out that the obligate putative obligate asexuals, deltoids, and the other ones are actually either facultatively or wholly sexual. Okay, so why are deltoid rotifers so important? Well, there's no evidence of males or meiosis. They're a monophyletic lineage. They're old. Some people might say ancient. I work on billion-year scales, so the literature calls them ancient. I'm okay with that, but they're 40 to 100 million years old, reasonably old. They're pretty speciose, so they don't have homologous chromosome pairs. They seem to mostly be absent. These retrotransposons, which are thought to proliferate through sex. And they have what's this sort of high levels of allelic heterozygosity called the Meselson effect. I'm going to tell you about that in a minute. This is the Meselson effect. So the idea is that, okay, and this is proposed as a way of diagnosing asexual eukaryotes. So if you're a sexual organism, and I go into any of your genomes and I pull out a locus and I look at both of your alleles, they're mostly going to be the same. They might differ by 1 or 2 nucleotides at most, right? So that's because we have all arisen from sexual reproduction, right? Because there's recombination going on all the time. And so divergence between alleles, big A and little a, is actually minimal within a species. However, if you're an asexual and you lose sex, then your big A and little a alleles are no longer recombining, no longer talking to each other, and they're going to diverge from each other from the point of asexuality. Okay? That's the Meselson effect. So that process was ascribed to deltoid rotifers, and it turns out that they actually have high, what we call allele sequence divergence. But it turns out another way of generating that same kind of signal is by degenerate polyploidy. Okay? So if the origin of the organism is by a polyploid and they're segregating out different copies of alleles that come from different sources, that you can actually have the same sort of apparent look. And it turns out that deltoid rotifers were originally thought to have this, and probably it turns out the reason is because they're degenerate tetraploids. So that piece of evidence sort of goes away. So the genetic evidence suggesting ancient asexuality is based on the consequences, but you can see where I'm going with this, but do deltoids have the capacity for sex? Right? So obviously the approach is we search deltoid genomes for meiosis-specific genes. If they're present, yes, there's something going on, they're doing it covertly, etc. Or if not, that would be consistent with ancient asexuality. This is now my first time I get to talk to you about degenerate PCR. So I sort of talked earlier on, you might have thought that we were only looking in genome sequences. It turns out that degenerate PCR was actually one of the major methods in my lab to look for these genes in lots of organisms. And this is a quote from my postdoc, easier said than done. So we can make alignments, we can make primers, and then we can go looking for these genes in lots of different organisms. So this was actually deployed in my lab. So here's now the army of people that did it in deltoid rotifers. It's actually a super hard thing to do. So anyway, so that's at various undergraduates, grad students, postdocs, et cetera, that were involved in this project. And I'm going to tell you about some of the results. So we didn't have genomes. Deltoid genomes were pretty big at the time. Now we have one that's been sequenced, which I'll tell you about. But we only had this approach, right? We had to target going looking for these genes. Okay, so here's RAD51. Now this is not meiosis specific. It's sort of our positive control. Okay, so we have monogononts, we have some deltoids. We go looking for the genes by degenerate PCR. We mostly have them, right? So we can do this. It works. Some of them have multiple copies, which is kind of maybe interesting. If we map the intron positions on them, they map to the same spots, so that gives us confidence that they're the same ortholog. We do the phylogenetic analysis. Great, they group in the right way, et cetera, et cetera, et cetera, right? So this isn't a big surprise for deltoids because remember, RAD51 is involved in the double-strand break repair as well, okay? So it's not meiosis specific. All right. That was just a tease because here's what happens when we look for DMC1, the meiosis-specific paralog. So we found it in monogononts, and it was kind of pretty hard to find. And we found it in deltoids, but the intron positions don't match, and it was super hard to find. And we did the phylogenetic analysis, and they kind of branch together, but there's not much taxon sampling going on. They're really long branches. It's sort of hard to know whether they're bona fide DMC1s. They might have been. So now we're not going to fill out this table for deltoid, for rotifers, and so everybody's got RAD51. We're not sure whether DMC1 is the right thing. So fast forward through a bunch of those, and note that we now have a lot of gray and a lot of missing data in the deltoid rotifers. until we get to MSH5. You've already met MSH5 a couple of times today. So monogonons, okay. Turns out we had a little bit of a hard time pulling it out of the monogonot genomic library that we had, but we pulled it out of genome sequencing that was also ongoing. So we got a couple of pieces from genome sequencing. Turns out we had really good luck pulling it out of deloid rotifers. And now, instead of showing you the triangles as introns, I just have the exons in alternating colors. So you can see not only, in different pieces, et cetera. So, basically the intron positions also match, unlike the previous one that I showed. So now we do the phylogeny, and then they look really good. So, okay, I will emphasize, MSH5 is meiosis specific. Deloid rotifers have MSH5. Okay? So this was our first piece of evidence that they've got a gene that is meiosis specific in everything else, and deloid and monogonot rotifers have it. So it's present. We actually extended this quite a bit. It's present and intact. Okay. So turns out that MSH5 is present. Turns out that MSH4 is also present. I'm going to show you in a minute from the genome. So we didn't have a lot of luck with this sort of degenerate PCR approach. So what was interesting was that when I first started doing this work, we were pulling out a lot of these genes from deloid rotifers. So up here on the top, I have green boxes on them. And I was going around to meetings saying, oh yes, they've got meiosis-specific genes. Until we did the phylogeny, and not so much. So it might be that they're contaminants. It might be something else. But now we're a little bit more cautious. But the answer is that they clearly have MSH5, and now there's a genome sequence, which I'll tell you about right now. So this genome sequence came out in now almost 2 years ago from Adenita vaga, and Adenita was one of our organisms that we worked on as well. So here's our MSH5. Right, we actually had pulled out MSH4 as well from PCR from this organism, and it turns out what was important is that MSH4 and MSH5 also work in a heterodimer, so you have one and you have the other at the same time. It turns out that in the genome they also found a copy of SPO11 and a copy of HOP1. Okay, so they've got 4 of the meiosis-specific gene. Now SPO11 is quite diverged, but it's recognizable as SPO11. So those are the data, right? But notice their title: Genomic Evidence for Ameiotic Evolution in the Deloid rotifer Adenita vaga. But yet they've got 4 of the meiosis-specific genes. So here's a quote: Our analysis of the lineage reveals positive evidence for asexual evolution. Its genome structure does not allow pairing of homologous chromosomes and therefore seems incompatible with conventional meiosis. I'm going to show you what their genome assembly looked like. So here would be a sort of standard genome comparison, right? So you got a diploid, you got homologous lining up, but it turns out that their genome is highly rearranged. So they took this as evidence. It's hard to imagine, but biology sometimes is hard to imagine that meiosis is actually able to work in this organism, right? Yet they have 4. They have 4 of the meiosis-specific genes. So what do we know about how cell division works in deloids? Like I said, meiosis is apparently absent. Oogenesis, presumably, the research is pretty old. And it turns out we actually tried to do this in my lab. They're actually pretty hard to work with. So we really don't know what's going on. One hint is that they do extrude polar bodies, which is consistent with a meiotic-like division. So we don't really know what's happening. So I think this is one of those huge open questions that especially people that have kind of cell biological experience could really make a big impact on. Okay, so it's my last slide in this section, and then I'll sum up. So here's the summary, is that meiosis-specific genes are present. What's the significance? Well, maybe deloids are doing it. Maybe they have the capacity for meiosis. Maybe they have a weird meiosis, maybe like Giardia, right? Maybe genes might have non-meiotic functions, and this is actually given that there are 4 of them, maybe this is more tenable, right? So MSH4 and 5 are involved in, well, they evolve from mismatch repair, and it turns out they're involved in sort of binding mismatches in DNA. Maybe those MSH4 and 5 are doing another job. That would be super cool, but unclear, okay? There's still 4 genes to sort of come up with new functions for. And then third, which I alluded to, which is that maybe they're by horizontal transfer, although at least some of these genes are pretty clearly phylogenetically related to monogonant genes. Okay, so I'm finishing up. So I posed a bunch of questions, and I'm going to try to provide some answers. So when did meiosis arise? Early in eukaryotic evolution. Prior to LICA. But we have that big stem between whatever the archaebacterial ancestor is and LICA, so we don't know exactly when. How did it arise? Pervasive gene duplication. Maybe, although my data don't have any bearing on this, maybe at about the same time. I mean, they happen at the same time in that branch, but we don't know relatively when. Maybe it has something to do with sort of a genome duplication. And then finally, from what prior functions? A lot of those genes that I talked about have roles in DNA repair in their ancestral state and certainly in prokaryotes. And how has it subsequently evolved? So this is now sort of the focus, since the origin questions are significantly more obscure. Certainly there's been a lot of gene loss, some gene gain, which I actually didn't tell you about, and some more gene duplication. I'm agnostic on can meiosis be lost? Deloid rotifers, there's still a lot of work to be done. Can meiosis be lost? I don't know. I think that's a super important question because it bears on this question. What are the consequences of meiotic evolution for the maintenance of sex? And stay tuned to our work and many other people's work on this question, which I think is still a super important question in evolutionary biology. So thank you.
So that was pretty long. So you've only got about 4 minutes for
4
questions.
minutes. That's 4 questions.
So, short, to the point questions. Yeah.
Thank you very much for your talk. Right here? Yeah, okay. So there's no evidence at all for duplication in prokaryotes, right? Any evidence?
There is no evidence?
For gene duplication?
In prokaryotes?
Yes. I mean, you said that it's a eukaryotic thing, so there's no evidence at all for that happening in prokaryotes on the one hand, and I don't know if I'm posing the right question. And if so, then duplication is a transition, or was what was changed was like expression? If so, if there was any duplication, or if there's any evidence for duplication in prokaryotes?
So I think, if I understand your question right, is sort of so there is gene duplication in prokaryotes. That is a process of molecular evolution in prokaryotes. It's just much more pervasive in eukaryotes. Now, one of the reasons probably is the way that we handle our genes on chromosomes. And one of the hints about the gene duplication is that, which I forgot to say, I apologize, is that meiosis doesn't make any sense unless you're diploid, right? So diploidy had to come first, right? Because otherwise, why do you need it, right? So diploidy by definition is a kind of duplication. Now, if you, in sex, of course, you're recombining at every generation. So gene duplication as a mechanism of evolution is, I think, most people would agree with me that it's much more pervasive in eukaryotes than in prokaryotes. Okay, but and so what I was trying to say was that this particular process, there are a bunch of gene duplications that happened after the divergence from prokaryotes, but before LICA, okay? And whether that has something to do with the origin of diploidy or not, I don't know, but there's certainly something there that, that's worth thinking about.
One more very short question. You can ask that.
Hi, I was wondering how could be reflected that the fact that some genes are turned on and others are not present in incomplete or different types of meiosis during the evolution of meiosis. It could be imperfect divisions in cells.
So you're asking about the loss of meiosis-specific genes?
No, what are the consequences of absence of some genes in the way of meiosis could happen in some intermediate process?
Right. So for example, like in flies and worms, like in flies in particular, so Drosophila actually is sort of we know it's having sex, right? But yet it's missing a whole bunch of these meiosis-specific genes. It turns out that where we can actually look carefully, it turns out that there might be the birth of new genes that do it in a slightly different way, okay? So in Drosophila, actually, we actually know that. I haven't had a chance to talk to you about that. But there's this example I showed for MSH4 and 5 is that there's already a built-in backup pathway that we now know about. So in some cases, there's one pathway or another that can be deployed. In some cases, there's another pathway that gets invented. Again, we have 2 or so billion years of evolution to work with, right? Times the number of lineages, right? So there's lots of ways to skin a cat, as it were. And so it's those cases, like when you lose some sets of genes that seem really important, can you then go figure out sort of, what happened to those functions? And sometimes when we look, and this is not particularly, not a lot of cases, but sometimes when we look, we can actually find that, well, there's these other genes that are specific to the group that arose that do that function, yeah.
Okay, thank you very much.
Thanks.
Buenos dias. Works? Can you hear me in the back? Yeah, because I need some room.
For this next section, I have the pleasure of introducing once more to you Professor Yvonne Rosenstein from the Department of Molecular Medicine and Bioprocesses at the Institute for Biotechnology.
Thank you. Okay, thank you. In fact, I'm a surrogate for Mario Zurita, who couldn't come, so I will be very short and introduce him, Dr. Shaheen Takbash, and he obtained his PhD from Carleton University in Canada, working on molecular biology of viruses, and then he switched gears. Then he went to the Pasteur Institute for postdoctoral work on skeletal myogenesis, and he stayed there, and he's actually working on stem cells in development, and he has been interested on the regulatory gene networks that regulate how stem cells establish and regulate organs and tissues. And I think he particularly focuses still on skeletal muscle. So Dr. Takbash, thank you very much.
Thank you very much. I love short introductions. Can you hear me in the back? Okay, nobody's sleeping. Right. So I'd like to first join my colleagues in thanking the organizers, Juan, Berenice, and all the associated members, because actually when I was thinking of how this committee and this organization was set up, it had an evolutionary underpinning, because I'm going to tell you something about fail-safe mechanisms. And right from the point when we arrived at the airport, I noticed a huge family who took care of us. And each one was there, I think, as a fail-safe mechanism to make sure if one of them failed, another one would back up and make sure that we're well taken care of. We're given water, we're given coffee, we're given tea. We're really spoiled. So thank you very much. I greatly appreciated the effort that you've put in and to organize this very important meeting. So I'd like to have a confession to make. I'm not a developmental biologist, but I'm not an evolutionary biologist. I'm more interested in developmental biology, but we've stumbled into evolution. And I realized the importance of having a good data set. A good data set to make arguments about evolution. I'd like to give you two examples of how we've been misled by really powerful genetic tools that told us things about how a tissue is organized and how it could have been evolved, and that we got it fairly wrong. So one thing I'd like to focus on is fail-safe mechanisms. Now, when you think about fail-safe mechanisms, whether it's a gene or a circuit or a structure, you would think that the most vulnerable aspect would have a backup system. And it turns out that that's not really the case. There are genes out there that are very powerful, that don't have paralogs. You mutate them, you die. You die before gastrulation, or you die even before birth. And there are genes that are not that important, at least from our point of view, and they have backup paralogs. So there isn't any rhyme and reason. So this suggests that perhaps it has just arose, those circuits or genes or structures arose by duplications, and they got fixed into the population either by design or function. And I'd like to take up what Ada was insisting on yesterday on function, or they're just hitchhiking. And we need to distinguish what is hitchhiking and what is really function. So Eric mentioned that, Inaki mentioned also the importance of regulatory circuits, etc. I'd like to use one example. of a structure, and I'm not going to mention it anymore. I'm going to just use it as an example. And this is the extraocular muscles over here. Now, as you know, you can move your eyes freely. In humans, you have 6 extraocular muscles. In mice, they have 7 extraocular muscles. Now, if you're an engineer and you looked at this, okay, we don't want to criticize the intelligent designer who did this for us, but anyway, let's do so. If you're an engineer and you look at this, you'd say, well, you have an eyeball. You can use a tripod design. 3 muscles, perfectly situated as a tripod with a camera, and you can move your eye in every direction. But then you would say, silly you, because if you lose one of those muscles, you mess up your eye. You can say, okay, the tripod design out. Let's over-engineer this. Let's throw in 7 muscles. 7 muscles in mice, you get this really elegant design, and it turns out you mutate or you have a misfunction in one of those muscles, and you end up with strabismus. Your eye goes the other way. So even over-engineering did not compensate this simple tripod design. So, I mean, these are games that we can play. We don't know what the ultimate design is because we don't know what the truth is yet. And this example I use as a fail-safe mechanism for a structure. I like to focus in the beginning. I like to give you 2 stories. Fail-safe mechanisms in genes, transcription factors, and those that are playing in cells. And then in the second part of the talk, I'd like to tell you something about gain and loss of a structure. And you've heard about that extensively in this meeting, how are traits acquired and those that are not fixed, lost in certain branches during evolution. So what I'm going to be telling you about is skeletal muscle. To remind you, it's not smooth and it's not cardiac. It's skeletal muscle, so it's voluntary. That allows you to move. Most of the skeletal muscles are attached to bones, but not all of them. The extraocular, as I mentioned, is not attached to a bone. So I realize that there are not that many developmental biologists in the audience, so I'd like to give you a little bit of ammunition to understand the story I'll tell you. And I'd like to insist on the quality of the experimental data because that's really important if you want to think in terms of ontogeny or phylogeny. So skeletal muscle, as you see over here, is arising from mesoderm. And as you all know, gastrulation is an important part in your life. You have ectoderm and endoderm established, and then subsequently mesoderm is induced. So ectoderm and endoderm derivatives, it's mesoderm that I'll be telling you about, and it's skeletal muscle right here that I'll be telling you about. Now, if you were a designer, be it an intelligent or unintelligent designer, and you were going to design an organism, there are complicated functions. So one way to do it is to do it by modules. You take modules and then the Jacob and Monod toolkit, and then you can have local specialization of those modules. So you have to essentially design a limited number of modules and then specialize them in particular ways. So if we have a hypothetical chordate embryo after gastrulation, one way to divide it up is along the anterior-posterior axis. And in this design, this hypothetical animal can negotiate feeding and breathing in its anterior end and excretion of waste in its posterior end. Then we can divide up and have bilateral symmetry. Turns out the notochord goes up and down and divides up the axis, and the neural tube as well. And then we can have further specializations along the medial lateral axis, and then segmentation, what I was telling you about, modules that can have specialization, but then iterated up and down. And a good example of that are these green segments here, which are somites. They're transient structures. They exist for a few hours or a few days, and then they're gone. But they leave many tissues in their place. And it turns out everything from neck down in amniotes, our skeletal muscles are derived from somites, all of them. So we're talking about mesoderm that's on either side of the midline. So if you look at trunk mesoderm, there are plenty of different types of trunk mesoderm. Axial, paraxial, intermediate, and lateral. And these are the derivatives, and we're going to focus on skeletal muscle. So all skeletal muscle from neck down is coming from these structures, somites, which are paraxial mesoderm. In the head, it's a little bit more complicated because this structure, this mesoderm, is not segmented. And you have at least two types of mesoderm here. Precordial and then paraxial lateral, which is combined. And skeletal muscle can arise from all of them. So what's highlighted in red is the tissue I'll be telling you about going through one root from neck down and multiple roots in the cranium. Now let's go back to more generalities and talk about chordates. Chordates, according to popular belief, have four important signatures. A midline notochord, hollow dorsal neural tube, nerve cord, pharyngeal pouches, which eventually have given rise to jaws, and a postanal tail. So this is, I'm representing, let's say, an amniote. If we go to an earlier stage, a tunicate, which is a urochordate that has many of the features of higher vertebrates, you notice in the embryonic stages, it does have a heart, does have a pharynx, pharyngeal pouches, and it does have a siphon. This structure has a notochord and dorsal neural tube. So this is an interesting organism to look at for homologies of characters. And I'll be coming to that organism at the end of the talk. Now, a lot of our understanding of how a tissue is established comes about from experimental embryology. But before I tell you about that, I'd like to mention something about the head, evolution of the head. So I'll leave our politicians out of this. Most morphological and functional differences among chordates occurs in the head. And the vertebrate head, it turns out, is largely neural crest derived. And neural crest are these specialized cells that in a way take on the role of mesoderm in the body. And they can make connective tissue, cartilage, bone, dermis. And it was thought at one time they also make muscle, but that was experimentally proven to be untrue. So in any case, many of your structures are established from neural crest. And Marion Borner-Fraser had speculated that this transcription factor, AP2, the acquisition of it in these migrating non-neural ectoderm cells resulted in the emergence of a neural crest in vertebrates from a protochordate. So as I mentioned, a lot of what we know came about from experimental embryology. And before the knockout technology came out in mice, we've been using model systems, C. elegans, fly, xenopus, etc., and they've told us a lot. And one important experimental model system has been the chick-quail chimera. And this is really through the driving force of Nicole de Duran in France, who discovered that if you take quail cells and if you take chick cells, their nucleoli are very different. So she exploited that by looking at the quail nucleolus, which is very large, and eventually generated antibodies monoclonal antibodies that can detect quail cells. And the experimental design was to take, and this is really a spectacular feat, to take bits of a quail and transplant them into a chick. And this exploded experimental embryology after that discovery. And so here I'm showing a somite. So somites give rise to all skeletal muscle, as I mentioned. Transplanted from a quail to a chick, and then you look at those derivatives, and then you can say where the limb muscles come from, where the neck muscles come from, etc. In this case, the neural tube was transplanted from a quail to a chick, and the neural crest, the dark feathers, you can see this animal went to birth, and the neural crest derivatives are coming from the quail in a chick chimera. This looks spectacular, but to me, actually, the most spectacular experiment that her group did was back in 1988 when they took the mesencephalon and diencephalon from a quail, transplanted it into the chick. And as you know, quails and chicks have distinct singing patterns. And it turned out that she ended up with a chick with a quail song. So that tells us that experimental embryology is powerful. It can give us not only the lineage, origins, but also how different traits can be transposed among similar species. So I'm a mouse embryologist. I'm going to be telling you about mice and the genetic tools that we use to look at fail-safe mechanisms and loss and gain of a structure. Now, to set that up, I'd like to tell you something about some core transcription factors. And this particular set of transcription factors was quite important. Back in the 1980s, Hal Weintraub's group discovered that a single gene transfected into a non-muscle cell converts that cell into a muscle cell. And that was MyoD, this transcription factor. And that exploded the field of molecular biology, and everybody started to look for tissue-specific master regulators in the liver, in the heart, etc., and among species, also in Drosophila and Xenopus. And they failed miserably. Nobody else was able to identify another gene that had such a powerful property to convert a single non-specific cell, fibroblast, or even a neuronal cell, into muscle. So this was curious. And then the knockout technology came out in the 90s. And meanwhile, other related transcription factors were identified. And it turns out if you do a triple mutant, if you knock out all of these three genes, you end up with mice without any skeletal muscle at all. So what happens in these animals? Well, actually, you don't need skeletal muscle until you're born. Until you're born, you need to breathe and need to swallow. So these animals go to birth, then they die. So this experiment told us, erroneously, that these transcription factors are probably working in the stem cells of that tissue. Why? Because if you remove these transcription factors, you don't make the tissue. And what makes the tissue? The stem cell. Turns out that was a wrong conclusion. They are in the daughters of the stem cells, and I'll show you a lineage scheme in the next slide. And as time went on, other transcription factors were identified. I'm going to mention this one later on, TBX1, acting in the head. So another point I'd like to make in passing is that life was not all bad. Everybody looked for these master regulators, and there were some evidences that came out in literature until Yamanaka, of course, did his key experiment that won the Nobel Prize, taking 4 transcription factors and even 1 transcription factor into a fibroblast can remove that fibroblast identity and shift that cell into an induced pluripotent cell, an embryonic state. So those transcription factors are clearly powerful, and it's a combinatorial of transcription factors that was necessary to change that fate. And now everybody is doing these kinds of experiments, and it's not a single gene necessarily, or it's a single gene in the right context, single transcription factor in the right context, that converts a fate from one fate to the other. So I just told you that if you knock out these genes in mice, you eliminate the tissue completely. So imagine a mouse without any skeletal muscle at all. This experiment could not be done in the chick because, for the time being, there's no genetics in chick. But there is genetics in C. elegans and in Drosophila, right? So if you take out the ancestral homologue of this, would you eliminate completely skeletal muscle in those organisms? It turns out that that's not the case. If you remove maternal and zygotic MyoD homologue, which is called Nautilus, you end up removing only a subset of muscles, but all the other muscles are okay. And it turns out that Drosophila has another design. It's modular, and it uses this MyoD homologue, Nautilus, in one set of muscles, but other sets of muscles are specified by other transcription factors, Kruppel, etc. So here, the vertebrate design, we would imagine, has used one of those modules, the Nautilus module, and expanded on that, and has eliminated the other signatures. In the case of C. elegans, the homologue for MyoD is called HLH1. If you mutate that, the body wall muscles are still present, but they're defective. But you don't eliminate them. So somewhere along the way, the vertebrate amniote design picked out one module and put all their eggs in that module and eliminated all backup systems. So another final point I'd like to make on this homology aspect is what is the driving force to keep these transcription factors? And it's a question I'd like to address. Why do you need 3 of these transcription factors? If you eliminate any single one, the muscles are grosso modo okay. You need to eliminate all three of them. So why do you need three identity genes in this one animal? So if you look at the selective pressure, if you compare human and xenopus myodi within a highly conserved region, they're highly conserved from human to xenopus. If you do the same thing for MIF5, mouse, xenopus, and human, they're highly conserved. Now if you take human and human, MIF5 and myodi, they're not very highly conserved. So that's telling us that throughout evolution there's been a selective pressure to maintain myodi as myodi and MIF5 as MIF5. And where do these genes sit in a lineage progression? As Eric was saying, development is moving forward. You're starting from a stem cell, you're ending up with a differentiated cell, and these transcription factors that I've been telling you about, these identity genes, are sitting in the daughters of the stem cell. And there are other regulators regulating the stem cells. So at this progenitor stage, the cell specified enters into another state. These transcription factors come on. If you make a mutant, they don't come on. You don't make the program. So you make a triple knockout, no muscle, and eventually this stem cell population is lost. Why? Because the daughters keep the mothers alive. The daughter cells, the progenitors, signal back through Notch delta signaling, that's the specialists, and keep the stem cells alive. If you remove the daughters, the stem cells die. The whole system collapses. So, coming back to the question, why do we have three cell fate determinants? So here's a scheme. As you know, skeletal muscle is throughout the body. And here's a blue embryo where the gene LACZ, the enzyme beta-galactosidase, is produced in all areas where you have skeletal muscle. So all areas that you see are blue are what is represented in this scheme. I remind you, we have cranial mesoderm that's not segmented, giving the muscles in the head, and the somites, which are segmented, giving muscles in the trunk. So upstream genes, whether you're in the body or in the head, you have this signature, and the core three transcription factors are giving you identity. And the key is, why do we have these cell fate determinants? So one can imagine that during evolution, as this gene got duplicated from the Nautilus single or HLH in C. elegans, as the gene got duplicated into 4 copies, 3 of them being cell fate determinants, the duplication correlated with a duplication of the lineage. So that's an interesting idea. You introduce a fail-safe system. Either you introduce a fail-safe system by having 3 backup genes, you lose one, you still have identity, or you introduce multiple lineage, each of them expressing in a subpopulation. You eliminate one population, doesn't matter, the other one is there, keeps you alive. So that's one model. And of course, we know that these stem cells in the adult that are involved in regeneration are heterogeneous. I'm not going to go into that data. So that heterogeneity can be arising from the heterogeneity of these multiple lineages. So to keep the question simple, I'm going to get rid of MR4. Why? Because MR4, this gene is expressed only in the embryo, and focus only on MIF5 and myodi, and present you some genetics. So those of you who are not developmental biologists and are not familiar with recombination, I'm going to show you some slides where we make use of this pre-recombination strategy. Basically, these LOXP sites, which are inverted repeats, are recognized by an enzyme called CRE, recombinase, and anything that is between, any DNA sequences between these lock sites is removed, and you end up with one lock site. So you can put reporter genes, you can put resistant genes, etc. And this is how the recombination is resolved. So let's go back to the question. We have this progenitor population. We have these transcription factors. Are they acting in the same cell, or is the lineage duplicated? So to answer that, this is a parallel model and this is a serial model. One can test that experimentally. One can kill the population and ask, is anything left? How can we do that? We can put a diphtherotoxin gene. So diphtherotoxin is a potent toxin that blocks translation and kills the cell very rapidly. So if we put that diphtherotoxin gene hidden and blocked by a DNA sequence, all we have to do is remove that DNA sequence, have the toxin come into play, kill the cell. And this is where the Cre recombination comes in that I told you about. So you have a stop sequence, diphtheria toxin, and this mouse contains a diphtheria toxin in a locus called ROSA 26. Not important here, but what's important to understand is that it is expressed in all cells. So all cells have this time bomb, diphtheria toxin, sitting in there. When the Cre comes in, removes the stop sequence, kills the cell. So we have a genetic strategy to kill the cell and ask if this model is operating or is this model operating. Well, it turns out that this cell ablation strategy was done by putting the Cre recombinase in this gene, MIF5, by two independent labs using two independent alleles of MIF5, using two independent diphtheria toxins, and they got the same result. All muscles were normal. So the experiment was taking MIF5 expressing cells, kill them, and see what happens. All muscles are normal. So that tells us that there must be a second population. Right? That means that this model is right. There are two parallel populations. Now this result influenced greatly our thinking in the field because it told us that perhaps gene duplication of these identity factors correlated with duplication of the lineage. So it happened that we had those experimental tools in our lab. And we did the same experiment. And we took both of these Cre-expressing MIF5 mice, crossed them to this time bomb, diphtheria toxin, and found, you can't see this, but please believe me, you've found that muscle groups are missing. And if you look at these animals, their paws are dropped. And that's a classic phenotype of lacking muscles in the limb. So this put us in a terribly uncomfortable position because, as you know, when you're coming in second, you have mountains to climb to prove that you're right. So one thing I'd like to point out is this number here. Fortunately, Glenda, who was a postdoc who did this work, she looked at 55 animals, and all 55 of them had this phenotype, muscles that are missing in the limb. So that's a reasonably sufficient N value. But this result is still troubling because you'd like to have results that are either on or off. This result is partially pregnant. It's not pregnant and it hasn't delivered the baby. It's somewhere in between. And it's an awful type of result to deal with because you ask yourself, why are not all the muscles gone? And why do you have a partial phenotype? So some muscles are still present. You see that? Some muscles are still present. And not all of them are gone. So is this a problem with the ablation? Now, as an experimental biologist, when you're doing this kind of design, one absolute criteria is that 100% of the cells should be killed. Right? Because if you do not kill 100% of the cells, and you see this population come up, they could be coming out from escapers that didn't get eliminated. And the second question, why are all muscles not ablated? Well, this is a little bit technical, but please bear with me, just look at the colors. So, you have two copies of this gene, MIF5. One of them has your Cre recombinase to unlock this time bomb and kill the cells. In the other copy, we can put like Z, right? And see all cells that express MIF5. So, if we do that, then we should see no blue embryos. We went a little bit further and put two other reporters, and this is basically your miner's canary. If the oxygen level is going low, canary dies, you better get out of the mine. And these are reporter genes telling us that the genetics is working properly. So basically, under these conditions, if you have a control where you just put in the Cre recombinase, it should be blue, it should be red, and here there's a tomato gene, it should be red. This is blue, blue, and red. If you come in and ablate this population that's expressing MIF5, you should have no blue cells, no blue cells, no red cells, right? So if the cells are dead, they should not be red. And when you do this experiment, you find blue, blue, and red. So this told us immediately that we do not have 100% kill. And so we have to question this two-lineage model. I'm going to go through this one very Thank you. So I have a dead battery. So let's look at now those experiments that I told you about were done in 2008. And of course, tools become more and more sophisticated as you go along. And this particular line, ZAP, is rather inadequate. And that was used at those times to tell the experimenters if their experiment was working properly or not. And I'll just show you some experimental evidence. Here's tongue, muscle, in control and an ablated. So if you look at this result, with the genetics that I told you, we've ablated the lineage, you should see no muscle there. They used a reporter, and they see no blue cells. That indicator, no blue cells, is telling you that you've eliminated all the cells that express MIF5. And you see this muscle here, you say, okay, it must have come from a second population, because we eliminated all cells that are expressing MIF5. That's our minus canary. That is an old generation mouse. If you look at the new generation reporters, you have red cells, you have blue cells, and they have not eliminated the population. So this told us that, in fact, thank you. This told us, in fact, that there was a problem with the reporter mouse that was being used. Now, that's not the killer experiment. Let's pose the null hypothesis. Let us assume that there are two separate lineages. We kill one with MIF5. The other one is being run by MyoD. Let's kill MyoD. If we kill MyoD, we should have no muscle, and the muscle and the lineage should be eliminated. So that experiment we did, let's assume that the two-lineage model is valid, expansion and rescue, and if you kill one population, and if you eliminate genetically MyoD, you should see no muscle at all. Thank you. That just died too, I think. Can you hear me? Yeah? Okay. So here's the model where we eliminate the one population. There's residual muscle there. Now, is that residual muscle due to the second lineage or escapers from the first lineage? How do we answer that question? Eliminate genetically that second transcription factor, MyoD. And if you do that, you eliminate MyoD, you still have muscle. So the two-lineage model is incorrect. And what we were looking at, or what they were looking at, is escaper cells masquerading as a second lineage. So I'll summarize these points that I just told you. Diphtherotoxin ablation, now this strategy is being used extensively to look at different populations in the nervous system, different populations in the intestine, etc. And if you ask this question, I remind you, you have to have a 100% perfect kill. And having 100% in biology is very difficult to do. So we would say that escaper MIF5 cells that were not eliminated are masquerading as a second lineage. I'm going to skip that point. And we favor a single progenitor cell lineage. In other words, this is incorrect, this is correct. So how does that influence our thinking? Well, instead of this model, having two lineages arising as the genes duplicated, we have, thank you. We have a single lineage. So how do you introduce the fail-safe mechanism? You already know that from experiments, if you believe what I told you, that you have a single lineage, but you have two transcription factors, even three, that are acting as a fail-safe mechanism. Well, it turns out we know enough about the enhancers of these to know that the signaling pathways that are acting on those enhancers have differential effects. So here's one way to introduce a fail-safe mechanism. You play with the enhancers. You have one responding to Wnt signaling and the other one not responding a little bit less, and then you have that switched or differentially sensitive. and the other transcription factor. So if something goes wrong intrinsically or extrinsically, you can still have a backup system that would activate the gene and keep that lineage in place. So this is what we would argue, that the fail-safe mechanism and the design is built into the enhancers, not in duplication of the lineage. So I'd like to leave that point now and tell you something about the loss and gain of a structure. Now, as I told you, in the head, skeletal muscles are established from this mesoderm called the cranial mesoderm. And it turns out that in tunicates also, and in mammals, a single progenitor can give rise to heart and skeletal muscles. So from neck down, that does not happen. The two types of mesoderm are quite separate. You have skeletal muscles from one type of mesoderm, paraxial, and you get the heart established from the lateral mesoderm. Very distinct, separated very early in the embryo. In the head, that's not the case. You have transcription factors coexisting for the heart and skeletal muscle. One is downregulated, makes one, or downregulate the other, makes the other. So if we look at our cranial and trunk design, and we look at the heart with two components of the heart. The first heart field, which is coming from this root, and the second heart field, which is coming from this root, but has a sister that is giving also skeletal muscle. So this was discovered by Robert Kelly about 15 years ago and showed in the mouse, and then subsequently showed in the chick. So what does that have to do with a structure that I'm going to tell you about, the esophagus? So bear with me, and I'll walk you through the arguments. The esophagus is part of the anterior foregut, and of course it connects your oral cavity to your stomach. And when you eat, you're going to do that shortly, the food is going to go into the stomach and be digested. So the esophagus is communicating with the outside world through the oral cavity and your anus. And if you stay in the anterior region, you discover that this anterior portion, the design is quite interesting. There's a lumen through which the bolus or the food passes, and you have smooth muscle and skeletal muscle lining. And of course, if you're having some of this wonderful cream of spinach soup, you have to operate the epiglottis so that that doesn't end up in your lungs, but it ends up in your stomach. And the trachea and the esophagus are initially one unit that separate in the anterior region. And they're separated by the epiglottis over here that allows breathing apparatus and food to go down separate tubes. So if you look at this structure, the esophagus, the skeletal muscles from the esophagus are quite extensive, but they're arriving late. They start over here and they work their way down from the neck down. And because the esophagus is sitting in the trunk, the idea was that the skeletal muscles in the esophagus are coming from these segmented structures I told you about, the somites. All right? And there's actually experimental evidence showing that. So the esophagus muscles are claimed to be of Semitic origin. And again, as with the two lineage model, if you're going to make any evolutionary statements, you have to know the ontology before you go back and do the phylogeny. So as late as 2013, PAX3, which is a gene that regulates the ones that are coming from the somites, the muscles are coming from the somites, was identified as an important regulator for establishing the skeletal muscles in the esophagus. But we found, in fact, that the esophagus muscles are negative for PAX3. And that puzzled us, because again, the literature is telling us that it's coming from neck down, and we looked at an important regulator, it's telling us that that's not the case. So we turned to this gene that was mentioned yesterday, TBX1. Now you don't have to read all of this, just to know that TBX1 has mutants of TBX1 have cranial facial abnormalities and cardiac abnormalities in human and in mice. And if you mutate TBX1, you end up losing craniofacial muscles. So we looked at this important regulator of mesoderm in the head and discovered that in fact all the esophagus muscles are missing. What you see in red is smooth muscle that's left behind. And here's a wild type animal. You have smooth and skeletal muscle. You look at the TBX1 mutant, you eliminate that. So this puzzled us, and it told us that in fact people had it wrong. The esophagus muscles are not coming from the somites, they're coming from cranial mesoderm. And 100% of these animals are lacking esophagus muscles. So I'm just going to give you a couple of bits of experimental evidence, again, for the non-developmental biologists. Just please bear with me because I'm going to try to make a link between muscles established in this structure in mice, tunicates, and birds. So we went one step further and asked, okay, you have no skeletal muscles. Are there other regulators? In looking at wild-type animals, again, I apologize for the light, but I have a dotted line over here to indicate what is happening in the wild-type animals. There's this population of cells that is leaving the trachea and going towards the esophagus and depositing the muscles in the esophagus. And this population of cells is marked by islet. And islet is an interesting transcription factor because it regulates not only muscles, skeletal muscles, but cardiac muscles as well. And this raised a green flag in our minds that there's something interesting that's going on. And it turns out, if you look at TBX mutants, these wings of islet cells that are going to the esophagus are missing. So here we have a link of another transcription factor that is perhaps taking cells from this embryological origin to the esophagus and depositing the muscles there. And if you look in TBX1 mutants, this transcription factor is missing. Now let's look at a scheme. I told you about skeletal muscle in the esophagus. Well, in fact, not all animals have skeletal muscle in the esophagus. Some have it partially down, humans, cats, pigs, kangaroo, opossum, dolphins, monkey. Some have it all the way down to the stomach, goats, sheep, giraffes, yaks, etc., etc. But birds and reptiles have been suggested not to have any skeletal muscle in the esophagus. Now, you might be puzzled and you say, well, these guys swallow, so why don't they have skeletal muscles? Skeletal muscles are voluntary muscles. You swallow, you're taking the food down. How do these animals manage to get their food from the oral cavity to their stomach without swallowing? Well, they do have smooth muscle, and they have contractions of the smooth muscle. So then you ask the question, well, why bother making skeletal muscles in this structure if the smooth muscles can do it? That I don't have an answer to. But let's look at the origins of these animals. This is my only tree. Birds, as you know, evolved from dinosaurs, so they're coming off the reptilia branch. Here are mammals. So if we look at birds, in fact, this is the image that I just showed you. In the TBX mutant, you don't have these wings going to the esophagus. If you look at birds, they actually do not have these wings. So birds resemble mouse mutants of TBX1. They do not have these islet cells, and they do not have skeletal muscle in their esophagus. They only have smooth muscle, but no skeletal muscle. So we have a link, transcription factor link, by doing genetics in the mouse, telling us that there's a population of cells that is leaving the tracheal region, going to the esophagus, establishing the structure, striated muscles. If you do a mutation, you eliminate that population, and it turns out in chicks, they're behaving like mouse mutants that don't have this population. So this is the model that we have. Wild-type mice have smooth and skeletal muscle in their esophagus. TBX mutant mice have smooth muscle, but no skeletal muscle. And birds are like TBX mutant mice. They have the smooth muscle, but no skeletal muscle in the structure. And it turns out that birds and perhaps reptiles have lost this character. And why would I say that? Because if you look at U.N.A.M, the tunicate that I mentioned at the beginning of the talk, they in fact seem to have anterior foregut striated muscles. And I'll just show you that by illustrating this point. So birds have lost it. Let's go back to the tunicates. What's happening in tunicates? There's some lovely work that was done by Lionel Christian, who showed that if you look at tunicates, they're at the single cell level, there's a single cell that will give rise to the cardiac lineage, as in vertebrates, and to foregut striated muscles, right over here. Siphon muscles in the tunicate and the heart, and islet 1, the gene that I showed you earlier, is a regulator of this process. And here's a mammalian scenario. So what do we have, to summarize? In mammals, and in birds actually, Craniopharyngeal mesoderm in the head, giving rise to the heart, portion of the heart muscle, and skeletal muscle. So second heart field and craniofacial skeletal muscles. That also in birds. In tunicates, you have this cardiopharyngeal mesoderm that's giving rise to the second heart field as well, to the heart, but to siphon muscles, and this is regulated by islet 1. And if we look what I just told you, we have this branch point coming off, esophagus skeletal muscles, which we would say are the homologs of the siphon muscles. And it's also regulated by islet 1, and birds do not have this branch point. So we would say that there's a third axis coming through. Now this raises the question, obvious question, of why and what is the selective advantage of losing this among reptiles and birds. So I'll speculate a little bit, but first to summarize, I told you that skeletal muscles in the esophagus are of cranial origin. That's an important point if we want to look at the phylogeny and the evolutionary significance of it. We propose a third axis for cranial mesoderm. What was shown before, that craniofacial muscles and secondary heart field are coming from this one population anteriorly, and we show that the esophagus skeletal muscles are also coming from this population. So that's schematically shown over here. Now, what is happening in humans? Well, it turns out we talked to surgeons who work with patients, DiGeorge's patients, haploinsufficient for TBX1, this mutation that removed the skeletal muscle, and nobody's looking at skeletal muscles in these patients. And some of these patients, in the extreme cases, have problems with swallowing. So that's also an important point to get the embryology right, not only for the evolutionary aspect of it, but in the case of looking at patients. So from the evolutionary standpoint, we like to go back to lamprey, the protochordate Amphioxus, snake, shark. And finally, just as I wrap up, to speculate why would avians have lost this voluntary skeletal muscle? They're obviously managing to swallow. So one of the interesting points about this structure is that it can be distended, but to a limited extent. And we would speculate that striated muscles are keeping that structure, the esophagus, from extending extensively. And perhaps, and one thing that we're looking at with Shweta, who did this work, perhaps the forfeiting of mastication may have been associated with the loss of this striated muscle. So if you look at birds, they don't chew their food, they swallow. And if you swallow your food, you need to extend your esophagus quite a bit more than organisms that chew their food, macerate it, and then ingest it. So that's the highly speculative part of the talk, and I'd like to leave you on that note and thank the people who did the work. So the first part about the duplication of the transcription factors and duplication of the lineage, showing that there's a single lineage, was done with Glenda Kumar in the lab and with Ram Kumar, who left the lab. And the second part on the esophagus was done by Shweta, another postdoc in the lab, in collaboration with Robert Kelly. And I think I'll end there, and thank you for your attention. Was I on time?
Yeah.
Yeah, ready.
Okay. Yeah, thank you for your talk. I enjoyed it a lot. I was just wondering, perhaps I am a little bit lost, but why you don't have the same phenotype when you do the knockout of MIF5? Why do you don't have this?
So there are two types of things. One is the genetics to kill the gene, and the other is the genetics to kill the cell. So there are these three transcription factors, let's say two to simplify it, MIF5 and MyoD. If you make a mutation in MIF5, muscle is there. If you make a mutation in MyoD, muscle is there. So if you just do the single gene knockout, the muscles are there. you make a triple gene knockout, then you have no muscles. You kill the cell population, putting this diphtherotoxin in, you remove the population, and there's no muscle if you do it cleanly.
Yeah, but in your scheme, it was sequential, the action of these transcription factors. So
Yes.
will it be the same? Like if you remove one of the genes, you stop the cascade?
Well, that's an important point. I didn't mention that, and I didn't mention anything about the efficiency of the Cree. By the way, anybody who's making mice or genetically modified organisms, you do it on paper first, and then you go create your animal. Test your animal, okay? Because what you do on paper is not necessarily what you wrote on the paper, or what you get actually. So in this case, Cree was put into the MIF5 locus, but nobody actually tested to see if the Cree was expressed properly. And if you do an RT-PCR of the Cree allele and the wild-type allele, you find that the Cree allele is lower. That's the first point. The second point is that this gene is expressed highly in the embryo, but it's being downregulated during differentiation. So you want the Cree to be working, but the expression of the gene is going down, and you want to be working to kill your cell, but you're fighting two battles at the same time. If you put the Cree in MyoD, there's no muscle at all. All the cells are killed.
Hi. You mentioned that reptiles and birds lost the skeletal muscle in the esophagus.
Yeah, so we haven't directly tested ourselves, but the literature tells us of reptiles. We tested the birds in quails and chick, and they don't have the skeletal muscle in the esophagus.
Yeah, and this is controlled by TBX, which also controls the brain and the heart. I was wondering if these animals lost TBX altogether, or if...
In fact, that's an important point. No, chicks, avians do have the gene TBX, but that next gene downstream, islet, which is expressed in muscles and in the heart, is missing. The ones that are making these wings to go to the esophagus. In birds, that's missing. So the TBX, the upstream regulator, is there in mammals and in avians. But the next downstream gene, islet 1, is present in the mammals, but absent in the avians. And we're making the correlation. There's no definitive proof. We haven't done the knockout. But we're making the correlation that the loss of this population, marked by islet, going to the esophagus, correlates with the loss of the structure, skeletal muscles in the esophagus.
Hi. So you talk about this fail-safe mechanism at the level of enhancers. Is it known if this is a generalized phenomena or is just specific of this gene?
Well, there's a great deal of work coming out on enhancers, super enhancers, regulation during the lineage, etc. And I'm getting to be a believer that that's an important, not only in the promoters, but an important way where you can regulate the genes. Now, the enhancers can be proximal, or they can be 1 megabase away, like Sonic Hedgehog, which makes it very difficult to keep order. But then you'd have to think about looping. So there are people who are looking at those kinds of questions by doing 4C, 5C techniques to look at looping. But yes, there are examples out there of modulating the enhancer. In fact, even MyoD and MIF5 are some of them, PAX3, PAX7. They're the enhancers spread out all over, and modulation of the enhancer or mutation of the enhancer can lead to disruption of the activity or the expression.
Okay, yes, but I mean specifically of the fail-safe mechanism you proposed.
The fail-safe mechanism. So, sure, sorry. There aren't that many experiments. I'm looking at Eric. He must know that there's some fail-safe mechanisms out there. I can't come up with any examples as clean as this. In fact, no, I'll be speculating. But I think we should be looking larger. So a similar experiment was done in the nervous system by one of those groups. They took neurogenin 1, neurogenin 2, which are also basic HLH transcription factors that regulate the fate of neural precursors. And they used the same diphtherotoxin strategy and found that the population was still there when you eliminate one transcription factor. And I suspect that has to be looked at very carefully to see if it's not erroneously having escaper cells coming through. But in that case, I believe the enhancers have been looked at, and they're regulated differentially among neurogen 1 and neurogen 2. So one can speculate that, in fact, the fail-safe mechanism could be hardwired into those enhancer modules rather than duplication of the lineage. But there's no evidence on that yet.
More questions? Oh, over here, Simonetta.
So yes, thank you. I know nothing about that, but I learned lots of things. So the mouse that doesn't, as the mutant is viable? You can...
So that's a good question. In fact, we cannot make any mutations that eliminate those muscles but don't have other effects. And the TBX mutant is not viable. It has other effects. It has cardiac defects, etc. So we're trying to think of ways to just eliminate a subset of muscles and see if that's going to be... I mean, we would like to make a mammalian bird. So can we make a mammal without esophagus muscles and see how that's... I'd love to do that. But for the time being, we cannot ablate those muscles without having collateral damage in other subsets. Yeah. But what I could say is the first slide that I showed you with extraocular muscles, this is telling us that not all muscles are alike. If you look at patients, myopathic patients, the most severe ones, the dystrophic patients, who die in their early 20s in a wheelchair, they die because all their muscles fail. Their diaphragm eventually fails, and they can't move their finger. But the extraocular muscles continue to work. So they die, but the extraocular muscles completely escape the disease. So there are, from the disease point of view, it's telling us that the skeletal muscles are not all alike, and some of them are escaping, even though it's a genetically homogeneous individual, some muscles are escaping that pathway.
Do you think that there could be some fail-safe mechanisms that relied on some variations on these cell populations that could, in case the other systems fail, that could overgrow and then fulfill the function, but not in a context where you would have something like cancer or anything like this?
So from a cell population point of view, I don't think there is enough detailed information, clean genetic data to tell us if there are other examples out there. I would like to think that yes, you don't have necessarily to do it by death, killing a population. You can handicap the population in a particular way, genetically, by touching some genes. So that population is out of the game, and have the remaining population that would not be handicapped, that be positioned someplace else, subjected to slightly different signaling cues, and that would survive that particular event. I can't give you any specific examples now, but there are mutations that affect deep back muscles and ventral hypaxial muscles, and those would survive, where the other ones would be ablated. So, but on the cell population level, on the tissue level I can tell you, but on the cell population level, I don't think we have enough fine-tuned information to get to the mechanistic aspect of it.
Okay, but at least at the tissue level, we can imagine this situation when the overgrowth of these other types of cell populations could fulfill these missing functions, or...
Right, you can imagine that. In fact, as you were saying that, one interesting analogy came to mind. That's Einstein's brain, in fact. So for many years, people were trying to look at Einstein's brain and see what was the difference between his brain and other people's brain. And it turns out that, I can't remember the exact portion of maybe it was the hippocampus in the region. He had a portion of the brain that was missing, actually. And the adjacent region which is necessary for spatial temporal visualization, that's what you see in the London taxi drivers, that's highly developed, right, was compensating. So I would look at physical phenomena and relativity, and I would understand very little. He would look at it, and he'd be already looking at the next question, because that portion of his brain was slightly more enlarged. Now, The causal relationship has not been established, but the anatomical phenomena was there. So one can imagine for tissues, I mean, you can have ablation of certain muscles and you have compensations. In fact, that's done experimentally. You can remove certain muscles from the lower leg and adjacent muscles hypertrophy to compensate. And if you imagine that happening in the wild, the animal can continue to run away from its predator, for example.
Okay, we're just about back on time. Thank you very much.
Thank you.
Now I have the pleasure to introduce you Dr. Octavio Miramontes, who is member of the Institute for Physics here in the National Autonomous University of Mexico. He is member of the Department of Complex Systems, and also is associated to the Center for Science for Complexity. Thank you very much, Dr. Miramontes, for being here today.
Many thanks to the organizers of this event for the invitation to chair this session on the evolution of societies, where Professor Stuart West is our invited speaker. The emergence and evolution of cooperation and social behavior has been a present problem since the times of Darwin. Cooperation is everywhere, but ecological and evolutionary theories have been firmly grounded on competition. Cooperation is so common and overwhelming in nature that a simple turn of our head will spot it around immediately. Appearing in multiple ways and forms, it is so widespread, so much widespread, that it is puzzling why scientists were not willing to easily acknowledge its ubiquitousness and importance. Cooperation and social phenomena are present in humans and primates and in social insects, the common examples usually given. But it is also present in unexpected places such as in plants or bacteria, or even as emergent phenomena in artificial societies. Why are we so late in acknowledging this fact? What is the reason of so many years in which biology has lacked a good evolutionary theory of cooperation and social emergence? Charles Darwin was already aware that social behavior among animals was a fundamental and worrisome contradiction in the mere fact of the existence, for example, of ant colonies. How can natural selection favor the worker ant that has given up its individuality in the name of the public anonymous ways of the common? It was short after the publication of On the Origin of Species that a mainstream view emerged where social life was a sort of abomination, and that the ultimate goal for the living was an egoist, strong, and that the best fit individuals be passing their genetic material to future generations. But it seemed that has not to be this way, as loosely stated by S.J. Gould. Struggle is often a metaphorical description and need not to be viewed as an overcombat. It is we don't know how the very first living organisms and the ecosystems on Earth look like. However, it is known that the most ancient fossilized organisms were cooperative and social. They were likely bacteria. It is worth mentioning that present-day bacteria biofilms are well known paradise for the emergence of complex social interactions, and that cooperative phenomena among microorganisms, in fact, and cooperative phenomena among microorganisms. In fact, it is remarkable that the horizontal gene transfer was initially discovered in bacteria, and that this mechanism of gene transferring is now regarded as a whole new paradigm in evolutionary biology. In order for the HGT to work, there are at least one essential requirement, that the cells involved in the process come and stay together for a while, socially interacting. This is the reason why biofilms are so important in the early evolution of social life and cooperation. The finding of HGT has triggered many fundamental questions. For example, is the prokaryote diversity and evolution the result of the horizontal exchange of genetic material that allows for the sharing and incorporation of new encoding possibilities, that is, genetic novelties that are more accessible to selection? In fact, cooperation played a crucial role in the emergence of multicellularity, regarded by Maynard Smith and St. Mary as one of the major transitions in the evolution of life. It is also interesting to notice that both authors regard the origin of social groups, for example ants, bees, wasps, and termites, as another major transition in evolution. They, however, neglected that cooperation is implicated in most of his identified major evolutionary transitions, to the point that the opportunity to visualize cooperation as an extremely important force that drives biological evolution was missed. What, in my point of view, is a visionary and extremely important observation done by St. Mary and Maynard Smith, particularly in his 95 Nature paper, is the potential role played by biological complexity on those transitions. This view has indeed developed into the present fields of complex systems and systems biology that are making extraordinary contributions to help understanding the emergence of societies. We are now familiar with terms such as self-organization, criticality, complex networks, and so on when discussing the emergence of sociality. A modern view of evolution of cooperation started when, in the 60s, William Hamilton advanced his elegant mathematical formalism, now known as kin selection. In recent years, however, critics have stated that this theory barely holds, and so there is an ongoing debate on whether this formalism is actually a good explanation or not for the emergence of sociality. Notably, authors such as Martin Nowak and Howard Wilson have advanced alternative points of view that nevertheless have left the field in turmoil. Stuart West has been at the very center of this opinion exchange, And I'm very sure that he will enlighten us on where we stand right now on this issue. It is then my pleasure to introduce our next speaker, Professor Stuart West, or Stu, as he is usually known. Stu got a biology degree from Cambridge University back in 1991 and got a PhD from Imperial College London at Silver Park. In 1998, Stu became a research fellow at Edinburgh University and a professor of evolutionary biology at the same university in 2005. He stayed there until 2008 when he moved as a professor of evolutionary biology in the Department of Zoology at Oxford University, where he remains until now. Stuart West has received a number of honors and awards. He is, for example, the Vice President of the Society for the Study of Evolution. He got the Scientific Medal from the Zoological Society of London. He is a Philip Leverhulme Prize Award and a Chancellor's Rising Star Award from Edinburgh University. He is very, very well known because of his editorial duties. For example, he is an associate editor of a number of top-ranking journals such as Evolution, The American Naturalist, Journal of Evolutionary Biology, and Animal Behavior. Stuart West is, with no doubt, a worldwide recognized outstanding expert on the evolution of cooperation and social behavior. He is the author of an impressive list of research papers on top journals, and is also the author of very well-known books such as Cell Allocation by Princeton University and a co-author of An Introduction to Behavioral Ecology by Willey and Blackwell. When I was invited to present Stuart, the organizers were not aware that, in fact, Stuart and I many years ago used to spend hours having very interesting conversations about science while drinking the correct amount of beer at the Silver Park Students' Bar, because we were students, PhD students at the same time. Dear Stu, I'm very, very pleased to meet you again here at U.N.A.M. and have the opportunity to listen to your, I'm sure it will be your interesting talk, and be very welcome.
Hello? Can you hear me? Is that working? Okay. Thanks very much for that very kind introduction. And so, like a lot of the other speakers, like a lot of the other speakers, I'd like to just give a really huge thanks before starting to the organizers. So Juan and Berenice have done an amazing job. But also I really wanted to emphasize the jobs by all the other students that have been helping them behind, sort of sorting drinks, moving us around and everything, have also just been doing brilliant jobs. And an especially big thanks to my handler, Marlene, who played a pivotal role in making sure that I got some nice ceviche while I'm here. Okay, so I'd like to start by taking us back to Linda's talk on Monday night, where she was talking about these two different societies. Let me see. So yeah, and she was comparing the societies that she had been studying, Teotihuacan, which I think I've finally learned to roughly say, with Mayan societies, and how basically up at Teotihuacan, it was a much more cooperative society than Mayan societies. It was a more cooperative corporate society, less hierarchical. And explaining this variation between societies is exactly the kind of question that I'm interested in. However, if you're interested in trying to explain why some societies are more cooperative than others, humans are not a good study organism. Okay. For a start here, we've only got two societies we can compare. We could come up with a sort of infinite number of variables that differ between those two societies. You could make countless explanations for why they are. To try and get enough independent, different societies for comparison would be very, very hard. A much easier approach is to study animals, where there is huge variations in the levels of society. So these are meerkats, which I'm sure are familiar to many of you from various nature documentaries. And what you get in meerkats is is this ringing out there? Or am I just imagining that? It is. More center. Is that better? Okay. If it's giving trouble again, I might not notice, so sort of wave a lot or something. So. Okay, so meerkats live in groups of about 20 adults and their dependent young. And what happens is that the group is divided into a couple of dominant breeders, and then the rest of the adults tend to be relatively subordinate. So instead of breeding, what they're doing is they're helping the other individuals breed. They're babysitting for and looking after the young of the dominant pair. And this is quite a costly process. So typically, if you're the one babysitting at the nest for 12 hours while everyone else goes out foraging, your weight will go down while the weight of the foragers is going up. Okay. But not all. But at the same time, there's lots of species where they're not in cooperative societies. So this is a relatively closely related mongoose, also lives in Africa. And, it's a solitary individual. It doesn't live in cooperative groups. Okay. But then also, and so we could you might think of this as a relatively not social species. And of course, at the level of the animal individuals, it's not cooperative. But as the major transition approach emphasizes, cooperation occurs at all levels of society. So the various cells that make up this slender mongoose actually are a group of cooperative cells that make up the multicellular organism. And we can see this kind of cooperation at all levels of biology. You get cooperation between species. So this is an example of the legume rhizobia, which go into the root nodules of leguminous plants, beans and peas, and they fix nitrogen from the atmosphere and provide it to the plants. Again, so here we've got all the various cells cooperating to make up a multicellular organism. The eukaryotic cell, as we've already heard a couple of very nice talks, was formed by a symbiosis cooperative event. And the genome itself the ability of the genome to produce interesting organisms relies on cooperation between different genes on the genome. And the problem that arises from all of these situations is why do individuals bother to cooperate? From an evolutionary perspective, cooperation poses a problem. And the problem is, if you had a sort of group of relatively cooperative individuals, and then a little selfish cheat popped up, an individual that didn't pay the cost of cooperation but could benefit from the cooperation of others arose, we'd expect it, and it could pop up by mutation or migration. We'd expect that cheat, the selfish cheat, to do better and gradually increase in frequency over generations, okay? Even though it would lead to a declining average fitness of the population. And this is the problem of cooperation. How can we explain cooperation? Now, as was mentioned in the introduction to my talk, there's a bit of controversy about this in the literature, but I think we actually have a very good theoretical understanding of how cooperation can be explained, and that we can basically split explanations of cooperation up into two classes. And the first class is that if we imagine a cooperative gene or a relatively cooperative allele, that could increase its frequency in the next generation by increasing the fitness of the individual which it's in, okay? So if there was some direct benefit to cooperating. A classic example of this, which is familiar to humans, is reciprocity. You help someone in the hope that they will help you back in the long run. There, cooperation, by helping someone, it's actually helping you in the long run. Another example from one of the photos I showed you earlier is in the interaction between rhizobia bacteria and the leguminous plants in which they live. So, as I said, rhizobia live in sort of little nodules, or they move into the nodules of the legume plants, where they take nitrogen, they fix nitrogen from the atmosphere and provide it to the plant. Okay? Now, if you make an individual cheat, not cooperate, not do that, you can do that quite cunningly by giving them a sort of air mixture that's got argon instead of nitrogen,
the plant punishes them, it sanctions them. It cuts off the oxygen supply, and it stops giving resources to them. So here you have a kind of reward or policing or enforcing mechanism which can make individuals cooperate. So if you don't cooperate, the sort of costs of being punished for that are worse than cooperating. Okay, the second major class of explanations for the evolution of cooperation is that an allele or gene could increase its frequency in the next generation by helping other copies of it in other individuals. Okay? And obviously the most simple way you can do this is by helping relatives. And this is Hamilton's idea of kin selection. So whereby by helping a relative reproduce, you're helping pass your own genes on to the next generation just indirectly. And this is famously sort of illustrated and quantified in Hamilton's rule. And Hamilton's rule basically says that cooperation which is costly, so altruistic cooperation, will be favored if the cost of that cooperation to the individual performing it is outweighed by the benefit to the individual being helped, weighted by the relatedness between them, which is relatedness is a sort of statistical measure of relative genetic similarity. So it's a measure of how much you value the reproduction of other individuals. Okay. And so Hamilton's rule predicts altruistic cooperation between relatives and when the relative benefits and costs are sufficiently right. Okay. I should point out, actually, I don't mind questions as I go along, so if anyone has any questions at any points, do ask. Now, whilst emphasizing that all of these things are really crucial, the relatedness between individuals and then the ecological costs and benefits of cooperation, for the rest of this talk, wrong bit. I'm going to focus on relatedness and the consequences of relatedness for cooperation. And again, I'm emphasizing this isn't because B and C are unimportant, but because I don't have enough time to go into everything. And what I really want to do is I want to focus and look at one variable and see how it can have comparable effects across very different taxa, groups of organisms. Okay. And so the prediction that I really want to be testing is that cooperation is more favored when relatedness is higher. And eventually then I'm going to work on to how that fits into major transitions. Okay. So as I said, the first question is, does higher relatedness lead to more cooperation? Now birds are really good organisms for testing this. And the reason for this is because some birds live in cooperative societies. So for example, this little fluffy pom-pom thing is a long-tailed tit from Britain. It lives in little cooperative groups, sort of in an analogous way to the meerkat. What you get is you get some subordinate individuals who are helping other dominant individuals breed. Then other bird species don't live in cooperative groups. For example, this quetzal here, which I'm sure is familiar to everyone here. I have to admit, making this slide made me a bit depressed. In England, this passes for quite an interesting, exciting bird. And it just wouldn't here. And then it made me think that everyone in the audience probably thinks that in general about Britain. I could have pictures of the weather up here or food. And I've got to go home tomorrow, so I wasn't very happy about this. But okay, so if we're interested in comparing these bird species, what we want to explain is why do some species live in cooperative groups, and some in not cooperative groups? And does relatedness influence this? I'm not saying relatedness is all the story, but I'm asking how does relatedness influence this? So what influences the relatedness within groups of birds? So birds tend to live in little family groups when they're living in cooperative groups. And it turns out that the major determinant of relatedness in those groups is the sex life of those birds. Basically, if you have a female and she's monogamous, she only mates once, then all the individuals within a group will be, the sort of offspring will be full siblings of each other. So here you've got relatedness, and then against the number of mates that a female has. As females get more promiscuous, it will mean the individuals within a family, the siblings, gradually become less related. Okay?
Eventually, if a female's mating enough males, everyone will just be half siblings. So we've got the prediction here quite clearly then, that promiscuity should be bad for cooperation. This is one way of looking at the quote that E.O. Wilson once made, that sex is an antisocial force in evolution, okay? The more promiscuous females are, the less cooperation we should expect. And does that happen in birds? And yes, it does. So if we look here, this is data from about 250 species. What you see is That's still working? Yeah. First of all, the level of promiscuity is lower in cooperative species, okay? And higher in non-cooperative species. And if you do sort of detailed phylogenetic analyses of this, it does look like that transitions to lower promiscuity, so more monogamous mating systems, lead to transitions to cooperation. And in cases where cooperation has been lost from a species, it tends to be associated with transitions to more promiscuous mating. And also, even if you look just within the cooperative species, you see that they tend to be more cooperative, which is measured by the percentage of nests where you get these little subordinate helpers, in species which are less promiscuous. Okay? So just to summarize that so far, so that fits quite nicely with Hamilton's law. What we've got is we've got variation in promiscuity, which leads to variation in relatedness, correlating with variation in the level of cooperation. Okay. Now, one potential problem with this is it's, of course, observational comparative data. It would be nice if we can also do the same kind of thing experimentally. And we can do that by turning to bacteria. So a couple of decades ago, we used to think of sort of bacteria as living little lonely unicellular lives. But in fact, they engage in a vast number of sort of social, potentially cooperative behaviors. And the most common form of social behaviors in bacteria is the production of sort of some extracellular factors, some factor that's produced, released from the cell, and it does something outside. It maybe gets some resources, breaks something down, kills competitors, that can provide a benefit to the local population of cells. So it's providing a benefit to more than just the individual that produced it. And with this, the problem of cooperation again arises. So for example, if you imagine a population where you had these blue cells which are producing the little black dots that go out and do something good, they're paying the cost of producing it. And then you have these cooperator cheater cells, which are or potential cheaters that don't do it. You can see that then they can still get the benefit. So if these little cells are going out and getting some resource, they can still potentially uptake it and benefit from it. So the problem of cooperation can still arise. Okay. And so there's now increasing attempts to study these kind of extracellular factors in bacteria and see if they are really cooperative. Obviously, in many cases where they look cooperative, they might not be. And one that we've worked on a lot are siderophore iron scavenging molecules, which are basically, so just like something up here, the cell produces these little molecules, so bacteria are often limited by iron, especially when they're within hosts, we withhold it from them. The siderophores will acquire iron and then be taken back up by the cells to help them grow. And we've studied this for quite a while. One of the reasons we worked on siderophores as a potential social study system is that you can just, by looking at a cell, tell whether or not it's producing siderophores. So the ones that produce siderophores are green. So when you plate out the bacteria, the siderophore produces are green, and the mutants, which don't produce siderophores, which you can either make yourself or they can arise naturally in populations, are sort of a milky white. So you don't have to do anything fancy to follow a population and see how cooperation or cheating is doing. And so we've done a number of experiments where we started with about, for example, one thing you can do is you can test the role of relatedness by just keeping them in population structures where they tend to interact with relatives or not. You can start with a 50-50 mixture of cooperators and cheats and see who wins over time. And the main result you get from this is if you culture them under conditions of high relatedness, a population structure that leads to relatives interacting, the cooperators quickly win. If you keep them under conditions of relatively low relatedness, the cheats take over time. Okay. So
I don't have time to go into this, but I wanted to very quickly mention it. Because I think this is quite an interesting example of how you can do something for purely evolutionary interest, not at all, but then you can notice that there are actually other implications of it. And so microbiologists refer to a lot of things as virulence factors, which are basically something when the bacteria produces it, it causes the host to die, or it causes damage or harm to the host. And in my experience, pretty much every time someone describes a virulence factor, what they're describing is a social or potentially social trait. It's a cooperative trait that's being produced and then released out into the cell. And so what this means is that you can actually see quite strong virulence consequences of the social behavior of these bacteria. So this is some experiments in mice where what happened, and this is the percentage of mice surviving, if you give them normal bacteria that cooperate, a lot of mice die very quickly. If you just give them cheap bacteria, which aren't doing things like producing siderophores or signaling or so on, they don't die so quickly. Well, they hardly die at all. But what's more amazing is actually this line here is a 50-50 mixture of cooperators and cheats, which the result of which for virulence is indistinguishable from if you just put in purely cheats. And so the amazing thing here is that as the social dynamics of the cheats coming in and out competing the cooperators and exploiting them is sort of totally dulling down the virulence effects of having those cooperators there, the normal sort of wild type cells in the population. And we're starting to sort of look at the longer term implications of this for clinical patterns, and also in fact whether you can actually use it, put it to some use. So for example, could you use the invasion of a cheat into a population infecting someone to bring a sort of medically beneficial allele into a population? Oh. But that's just a quick sideline. So just to quickly sum up the bird and the bacteria work and lots of other studies that other people have done in other things like mammals and so on, there's basically lots of experimental and observational evidence that high relatedness does favor cooperation. I think that's pretty well established now. You'd have to be mad to argue with that. But that's not, we're here to talk about major evolutionary transitions. And major evolutionary transitions aren't just about cooperation. As Rick was emphasizing in his talk yesterday, and I'm going to be following him in talking about the specific subset of major transitions that lead to a change in a new level of individual arising, so multicellularity, eusociality, some symbiosis events. This involves much more than cooperation. It involves a really extreme form of cooperation. And one way of looking at this is to split a major transition up into just two steps. First, you get a cooperative group form, so something like a meerkat or bacteria group would count as that. But second, you need group transformation, other things to go on to create a new higher level of individual. And again, as Rick was emphasizing yesterday, what you need is one thing is you need a mutual dependence between these different members of this new group. You can't split them up such that they could reproduce on their own anymore. So they're sort of mutually dependent on each other. You also need a sort of almost complete lack of conflict within the members of the group, such that you can think of that new group as a new higher level sort of individual, sort of as a single maximizing agent. You can think of it as that rather than having to think of the smaller parts. So, and to me, the really exciting thing about the major transition approach was that it emphasized how these same problems, the same problem in making this transformation and all the steps that are involving mutual dependence, again, also as emphasized by Rick yesterday, often it will involve a division of labor between the different members of the group. And the really exciting thing was making clear that these same problems have arisen at these key moments in the evolution of life on Earth. So we've got the bringing together of a genome, the formation of the eukaryotic cell, the evolution of multicellularity, the evolution of eusocial insects, other between species mutualisms. And so we've got this lovely general picture that these same problems arise again and again. And I think the main thing that we have to do now is to try and work out to what extent can we give the same answers. If we can really give the same, if we can simplify and link the answers to these different transitions in the same way, then we can end up with an incredibly simple and elegant description of how complex life on Earth has evolved. Okay, and so what I want to do for the rest of this talk is just quickly focus on two transitions and just the role of relatedness in them. And that's the transitions to multicellularity, as illustrated here by this multicellular agouti, and the transition to eusociality, as illustrated here by this sort of soldier from a leafcutter ant. And so first I'm just going to talk about multicellularity. And so depending on how it's defined, multicellularity, you can argue, has arisen about 25 times. But it's led to very different levels of complexity. Sometimes you get quite exciting, complex things like this agouti, and sometimes it leads to things like slime molds, which they look nice, but they're really not that complex. This is quite a difference. How can we explain when this happens or when this happens? And how important is relatedness in explaining that? Does relatedness help us explain it? And again, as before, so we again need to say, okay, what influences relatedness in this case? And as Inaki mentioned in his talk yesterday, multicellular groups, multicellular species, form their multicellular groups in two very different ways. One is you get cells staying with the parent cell that created with them, and this leads to sort of a clonal group, okay, so where we've got relatedness equals 1 between the cells. I've illustrated this here with the human I'm illustrating this multicellular group with is Bertie, my PhD student, who collected all the data for this work and did it. She's actually just submitted her thesis while I'm away. So I can't remember, I think this was being screened, wasn't it, I think? In which case Bertie might be watching, so hello Bertie, well done. But I also think you've probably got better things you could be doing than watching me speak. So anyhow, this is one way to form a group, which leads to clonal groups with incredibly high relatedness, R equals 1. The other way some multicellular groups form, such as the slime molds, is by aggregation, different cells coming together, okay? And the crucial difference here is that this leads to potentially non-clonal groups. It could, in extreme circumstances, lead to clonal groups, but in cases where people have looked, it doesn't. So we've got this way of group formation leads to clonal groups, that way leads to non-clonal groups. Okay, so we've got difference in relatedness following from difference in the way groups form. Oh. Now, a problem with testing these kind of influences of the effect of multicellularity on the influence of relatedness, or anything in fact, on the evolution of multicellularity, is it hasn't really arisen that many times, okay? And so we're always going to be limited in our power of sort of how many phylogenetically interesting contrasts we can make between species. But you can see, if you look at the species or the groups where we were able to get data, the little black dots, I don't know how well you guys can read this, this is sort of animals over here, and the black dot shows clonal group formation. You've got something up there, like cellular slime molds, where you've got an open dot, which is showing non-clonal group formation. And the crucial point from this figure is that you've got clonal and non-clonal group formation arising multiple times. So we've got a bit of phylogenetic power. And there's also some really interesting cases like the ciliates and the cyanobacteria where you have both types of group formation within a group. Oh. So what kind of things could we test influenced by relatedness? What will be our measure of sociality, our measure of complexity? The first obvious thing is, again, as Rick was mentioning yesterday, there's sometimes a division made between complex multicellular organisms, so which have a very early divide between germ soma, lots of different cell types, large numbers of cells, and that's happened 5 times. It's the animals, plants, fungi, and the red and brown algae. Now, we actually can't do anything useful with that. We can't test how relatedness or other things have influenced that. And the reason for that is it just hasn't happened enough times, and the times when it has, they're grouped together. You've got the red and the brown algae over there, you've got the fungi, yeast, animals there, and then plants up there as well. So you've got these sort of 2 clumps where it's happened multiple times. So there's just not enough phylogenetic power here. It's like having only n equals 2, essentially. So we can't do anything interesting with that.
But instead, what we can do is we can split down into the various things that make up how complex a multicellular organism is. And in particular, I want to talk about three things that we can measure and quantify and that vary enough across species. And I'm going to illustrate them here with this cyanobacteria species, Anabina. The first thing we can ask is how many different cell types do they have? We have a lot of cell types. If you look at a cyanobacteria, some species have just one cell type that does everything. They've got no division of labor. So again, like the Volvox Rick was talking about yesterday, but there's variation in the amount of division of labor. In other cyanobacteria species, you do get division of labor. One common thing is you get some cells that fix nitrogen, called heterocysts. They divide the nitrogen fixing between those cells and then the reproducing cells. The advantage of that seems to be that the oxygen is basically really bad for nitrogenase. Other divisions of labor you can get these kinates, which are spore cells popping up, almost sort of analogous to spore cells that can survive harsh conditions. In some cyanobacteria species, you can get other types. You can get a sort of dispersing type, which is called a hormonogonia. But crucially, this varies across species, the number of cell types. We can count it for us, we can count it for other mammals, we can count it for cyanobacteria. The second thing we might ask is, do you get sterile cells arising? So cells that are purely altruistic, just benefiting others. So again, Rick was discussing this kind of thing yesterday with respect to the germ-soma divide. And an example in the cyanobacteria is these little heterocyst cells that fix nitrogen and don't reproduce. The third thing we could measure is group size. How many cells do you get? How big does your group become? And so these three things are sort of various measures of the complexity of the social group. It's how much division of labor do you have, how altruistic is your society, and how big is your society. And what we can do is we can compare this between clonal groups and non-clonal groups and see if there's a difference. And it turns out, luckily, there is. So first of all, just looking at number of cell types, you get a lot more when clonal than when non-clonal. Second, probability of sterile cells. Again, that's just a lot likely more to happen when it's clonal than when you're non-clonal. And third, number of cells. So group size is on average about 100 times bigger when you're in a clonal versus a non-clonal group. But this result isn't actually, despite this huge effect, it's not significant. And again, I think I suspect this effect is really strong, but it's just limited by the sort of the phylogenetic statistical power we have here for this analysis. So, just to summarize that a little bit, what we have then is that higher relatedness, a way of group forming that leads to clonal groups, does lead to greater sociality across multicellular groups. But again, I'm just talking about cooperation here really. I'm not speaking specifically to whether or not a major transition has been made. And if you remember when I talked about the major transition and the importance of group transformation earlier, following on from Rick yesterday, I was talking about the cases where you get mutual dependence between the different cells within a group, okay? And that's one of the key factors in whether a major transition in individuality has been made. So for example, in a mammal, like this rabbit, you can't split up the cells and then they go and live their life on their own, okay? A single cell can't complete its life cycle. And this is obligate multicellularity, has to be multicellular. In other species, they don't necessarily have to go through a multicellular phase. So for example, in this slime mold, Dictyostelium discoideum, they just go into a multicellular phase under certain environmental conditions when stressed to form a fruiting body. And even if they couldn't do that then, they would still be able to, they would do a lot worse, but there would still be some fitness. And so what I want to ask is, well, okay, how much does relatedness matter to whether or not you make this major transition to obligate multicellularity? And if you look at the data on that, it turns out that the transition to obligate multicellularity has only been made with clonal groups, okay? So we've got, considering the number of cases we had, sometimes with clonal transitions it goes to facultative, sometimes to obligate. With non-clonal, it only ever goes to facultative. Okay.
And there's some really nice examples here when I talked about earlier when we had the same type of, different types of group formation within group. So, for example, within ciliates, you get clonal ciliates have become obligate, whereas non-clonal ciliates haven't become obligate. They've only become facultative multicellular. And exactly the same thing going on in the cyanobacteria. So the crucial point here is that we can say, yes, generally, relatedness is good for cooperation, but actually, if you want to make a major transition, in this case to multicellularity, you don't just need high relatedness, you need a very specific condition. It's only ever happened under clonality. Okay. So, what about eusociality? I want to be able to compare eusociality with this. So how well can we compare the evolution of multicellularity with the evolution of eusociality? And exactly the same things arise. So in the eusocial insects, we have these different casts, which are analogous to different cell types. This example here is a Mexican honeypot ant, where some individuals, I don't know how well you can see this, they basically become living larders, just sort of hanging on the roofs with a huge extended abdomen that's just filled with a sugary solution to store food for when conditions are bad. I'd really like to see this species, and I haven't yet. So if anyone's got any with them, could they show me at coffee afterwards, actually? And the other big crucial thing here is, again, mutual dependence in the same way arises. We get obligate eusociality in the same way we can think of as obligate multicellularity. So this is a higher termite here. Again, not sure how well you can see this, but basically the queen is just a giant fat blob. She's an egg-laying machine. She cannot reproduce without the help of the workers. And in contrast, the workers are completely sterile. They cannot reproduce without the queen. There's a complete dependence between the workers and the reproductives. So, again, going back to the same question, so what influences relatedness on the evolution of eusociality? And this is quite easy for us because it's the same thing that mattered with birds, okay? It's how promiscuous are females, essentially. And if you look at the data on the social insects, so this just shows the Hymenoptera, but the same results apply more generally to all the other non-Hymenoptera eusocial insects as well. What you see is that eusociality has only ever evolved with strict lifetime monogamy. It has only evolved in species where females only mate once in that entire lifetime. Okay, so it's not just less promiscuity is good, you have to be completely monogamous. And so the phylogeny here, basically this shows different each of these different bars down here shows different evolutions of eusociality, except for the allodapine bees, which aren't. And black lines are complete strict monogamy. Now, multiple mating does happen in some social insects. So for example, if we look at ants down here or at honeybees, there's multiple mating. And red is multiple mating. But the crucial point here is that this is always a derived state. It is always evolved after you've got the different castes, you've got mutual dependence, you've got obligate eusociality. So you sort of can't go backwards. You've made that major transition. And this was really pointed out by Koos Boomsma. So for years, everyone had sort of wondered, well, maybe how important is relatedness? By pointing out that all these multiple mating ones are derived and that this has happened after obligate eusociality was fixed, he made clear the sort of complete fundamental role of monogamy. Okay. So, summary so far. Again, relatedness helps cooperation, but to make these major transitions to obligate multicellularity or obligate eusociality, we seem to need special conditions. We need clonality to get to multicellularity, or we need complete lifetime monogamy to get to eusociality. I say clonality down there because there's also eusocial aphids, which are asexual, and again, are doing it via clonal group formation. So I keep going to automatically move on. I'm not used to using a fancy clicker. So, but how can we link these conditions that favor multicellularity and eusociality? So they're both extreme conditions, but they're a bit different, okay? One is clonality, relatedness equals 1, everyone's identical. The other one's strict lifetime monogamy.
you share only half your genes with the other individuals you're helping. Is there any way in which we can sort of like simplify this into one simple story? And there is. And the crucial thing that you need to do this is to think about the sort of evolutionary options open to a potential helper in one of these societies. So a cell that's deciding that sort of could potentially help, or an individual that could help in a eusocial colony. And you have to consider the relative costs and benefits of the different evolutionary options that are open to them. Okay, and this is a sort of extended form of Hamilton's rule, making multiple partners more explicit. And what you could do is an individual could produce its own offspring, to which it's related by R0. So for example, clonal, in asexual species this would be R is 1. In a sexual species this would be R equals a half. And here you have to compare that with what they could do, the relative value of helping other individuals. And that's going to depend on how many other individuals they could raise by helping instead of producing themselves, and their relative relatedness to them. Okay. Now if we think about clonality, what you've got down here is 1, and what you've got across there is relatedness 1 as well. Okay. So in clonal groups, in multicellular groups, these relatedness terms drop out. So the amazing thing about that then is it removes any conflict over whether you should help or produce. All that matters is which option will be more economically useful. Okay? But then now if we think about a sexual species, you've got a half down there, but the amazing thing about strict lifetime monogamy is that relatedness to your siblings that you'll raise is also equal to a half. So the crucial thing here is you're equally related to the offspring you would produce yourself as the offspring you could help raise. Okay? So relatedness cancels out in exactly the same way with clonality in asexual species and strict lifetime monogamy in sexual species. So other conflicts can arise in different sort of population structures, but in these specific conditions, conflict is removed and over whether you should reproduce or help others. So it turns out that these extreme conditions, clonality and lifetime monogamy, have exactly the same evolutionary consequences for the evolution of eusociality, or of a major transition to the next level of individual. Okay, and so actually, we can think about the evolution of multicellularity and the evolution of eusociality in exactly the same way. So at this point, so what? So we've got that result. Does it mean anything extra? And I think it does, because I think what it does is it actually just makes the world a lot simpler. It really simplifies things for us. And I'm going to give 3 examples of ways that does. First of all, it means we can start making broad generalizations across major transitions. So I was asking, as I said earlier, I think one of the most interesting things is, can we say that the different transitions are solved in the same ways? And for eusociality and multicellularity, we can. We can say what's really crucial is group formation, and group formation is really crucial because of how it influences relatedness and removes conflict. And it leads to that condition where R offspring equals R of who you could be helping. Okay? So we get this, we can make broad generalizations across transitions. Second, I think in terms of major transitions, it tells us that everything isn't on a continuum. We can't necessarily think of all taxa as being on a continuum from not social to making a major transition, because making a major transition requires extreme conditions. So it requires clonality or strict lifetime monogamy. And if you're under those conditions, then given the right ecological benefit, you could make a jump to a cooperative society, or maybe even a major transition like that leafcutter ant there. But if you're not under those conditions, you're just not going to make that jump. All you can get to is a cooperative society. So for example, in the cooperative breeding birds and mammals, there's no surprise at all that they've never made a major transition, because there's always some promiscuity. There's always enough conflict within the group to stop that. And finally, I think it also means that when we're studying major transitions, we can actually just ignore and not worry about some things that we have to think about quite a lot when we're thinking about the evolution of cooperation. And an example of this is kin discrimination. So in lots of species, individuals will preferentially give help towards closer relatives. So in long-tailed tits, when they're choosing which nest to go and help at, they will go and help at nests of closer relatives, which they tell by sort of similarity in song. Or with this slime mold, this Dictyostelium purpureum, when they form these amazing sort of multicellular fruiting bodies, they will preferentially group together with clone mates to form those and sort of exclude non-clone mates. So kin discrimination can be very important for cooperation, but it's not going to have played a role in a major transition. And the reason for that is so that the conditions under which major transitions have taken place have got such similar relatedness that there's just no selection for kin discrimination. So for example, this shows some data here from cooperative breeding vertebrates. Up here, this axis as you go up is how much kin discrimination they have. So things like the long-tailed tit there are highly discriminatory. They really help closer relatives. As you go down, they're not bothering to direct help towards relatives so much. So for example, the superb fairy wren or kookaburra, they don't really help closer relatives. They don't show kin discrimination. And this correlates with the relatedness within a group. As everyone gets in a group gets more related, there's no benefit to helping closer kin because relatedness doesn't vary that much, so they don't do it as much. And where major transitions are happening is down at these extreme cases where there just isn't selection for kin discrimination. Oh, again. Okay, so just this is essentially my last slide. I just wanted to emphasize here. So as I said, I'd sort of gone into a kind of very specific case. We've got ecology and relatedness should matter. I've just looked at relatedness. I've just looked at it at two major transitions. And so I just really want to emphasize how much more work there is to be done here. So I think it is a really exciting time in terms of how much can we generalize across different major transitions. And I've missed out a lot of relevant things here. Is group formation equally important for transitions that involve endosymbiosis or transitions between species? Are things like repression of conflict or discrimination equally not important in other major transitions? And I think that's one of the major steps for the next few years, trying to work out if we can see if it is same answers to the same problems, in which case we really will be simplifying our view of the world. So again, and just a final thing to list all the people that have been involved in this work. Thank you very much, and thank you for listening.
I think we're good for time. In the straight through. Where? Sorry.
Thank you very much for a very nice talk. I would like to ask you plenty of things, but I will just ask you one question.
Okay.
In multicellular organisms, there is a social rebellion known as cancer. Do you know of any kind of social cancer in obligate eusocial insects?
Well, I guess it depends. It's going to depend on how you want to define it. So if you're thinking of a cancer in sort of social evolution terms, it's a mutation popping up that is causing individuals to cheat, essentially, to overgrow at the cost of the other cells around them. So when you get, in bacteria, things like individuals that don't do the social traits that we could think of as cheats will pop up and arise in a population. So you could think of that analogously. The dynamics of it are identical. In the social insects, you don't have mutations of as huge effect popping up. So for example, the siderophore scavenging molecules we work on in Pseudomonas, you can have a single mutation popping up that will completely knock out that trait. There isn't really an analogous thing in social insects. And there's another slight complication as well with cases where cheating is a real sort of evolved adaptation that is being favored and maintained in a population, as opposed to something like cancer, which I wouldn't think of as an adaptation. It's a mutation that pops out and then is with fitness consequences that's then lost.
You, and then you.
Thank you. I had a question. So now you centralize relatedness in the question of the major transitions. So as a conflict mediator, so if you're related, you don't get cheaters, and therefore you can make a transition towards like a new individuality. But aren't there many cases in which you look to, say, ancestors that have the same kind of relatedness but don't make the transition? So, which puts into question, even though relatedness is required maybe for the transition, it might not be a really good predictor for which lineages make a transition. And then the question is, which other ingredients do you think are actually important to understand as well?
Yes, so I mean definitely. So as I was emphasizing, it's when you've always got to look at the essentially the costs and benefits and relatedness. Relatedness is just a way of weighting those costs and benefits. And I think that's it. I mean, one thing I'm interested in the moment is how well we can generalize that across the different major transitions. For example, within the eusocial insects, you can divide them into two ecological reasons that seem to have favored eusociality, that have made the benefit to cost high enough. One of them is fortress defense. So you're defending, you've got a food resource in an area and you've got to defend it. That's social aphids, it's termites. The other major reason is a kind of, when you have a period of extended parental care and there's a reasonable chance that you'll die, the parent will die during that, it's quite efficient to group together to raise offspring. So for example, if you die and you've only raised half a larvae, that's completely lost if you're on your own. But if there's a few of you grouping together, then that investment isn't lost. So there's a sort of efficiency or insurance benefit there. And so that's well established for the social insects, that we can divide it into that. I think for multicellularity, you probably can as well. So for example, there can be a predation effect. So algae which clump together multicellularly to avoid predators, as opposed to species like the Dictyostelium slime mold, where it's increasing the efficiency of cooperation. By making a fruiting body, you're more efficiently cooperating and spreading. Or bacteria, by clumping together, they can make more efficient use of these goods that they sort of release out into the cells.
Hi. I hope I got this right, and I'm not going to make an incredibly stupid question. So you said that...
That usually means a very hard question is coming.
Oh, okay. So you said clonality it leads to obligate sociality.
Well, obligate multicellularity has only evolved when group formation is clonal.
Uh-huh. Okay, so...
Sometimes it leads to facultative, but you've only got obligate when it's that.
Okay, but this is related also to monogamy, right? Or not? Yes?
As in related to monogamy in the sexual species?
Yeah.
Yeah. So I think that's a really key thing, that they're incredibly related because they lead to a relatedness structure that removes conflict over whether to help someone else breed or help yourself breed. Mm-hmm. And even though they do it with different R values, they do it in an exactly analogous way.
Mm-hmm. So you used the rabbit to exemplify this, and I'm thinking that there is a phenomenon, at least in mammals, in which cells kind of stop being cloned as there is a mosaic of chromosomes. So you also showed this tree in which you showed that this promiscuity arises again. So I'm thinking if that might be related to that loss of, let's say, clonality because of this mosaic phenomenon.
Yeah, so I think that's a very interesting point. And I think what you're emphasizing there is that we have to make a distinction sometimes between the initial major transition to something that's obligate and then what happens after that.
So once you've made a major transition, evolution can lead to ways that reintroduce conflicts that weren't there before. And multiple mating or multiple queen in the social insects are good examples of that. And again, in multicellular organisms, yeah, you can start having enough cells. You can grow so big that mutation starts becoming important, which is something that Rick's modeled. Again, cancerous type things could be important. We've evolved genomic imprinting in mammals, which can lead to conflict, increased conflict between individuals. And so, yeah, and so I think those are separate questions. There's how did we get the original major transition? And then what happens after you've made it? Do new conflicts arise? Are they enough to break up the major transition? Or has it become so obligate that it can't be broken up? If they arise, how are they suppressed? And so on. So, yeah.
So I've got several eager questions over here, but I'm going to take advantage of is this a bully pulpit? I'm going to take advantage of my position and make a comment that you might want to respond to about the evidence for cooperation in bacteria. And that comment is that there is abundant evidence in bacterial cultures in the laboratory of processes that we can interpret as cooperative that fit into the public good structure. And those processes can be very useful models of the evolution of cooperation, as Stuart has shown, but there is remarkably little evidence in the real world of those bacteria that these processes actually serve a cooperative function. We have very seductive just so stories that seem to have largely overwhelmed what should have been a request for rigorous evidence of cooperation.
Yeah, I think that's a really good point, and I think it was true maybe 5 years ago. But I think one of the most important things in social microbe work has been to move towards trying to work in more realistic and in vivo situations. And so, for example, the data that I showed there from mice infections, one of the first steps that we did in that was to look, for example, to see these traits we've been looking at in test tubes, are they social in the same way in infections? And they are. And so, for example, you showed that with Pseudomonas in mice. People have again been looking at that with quorum sensing in Pseudomonas in infecting insects, not the natural hosts, but a step up. If you look at people who've been doing stuff with staph on that, there's some really nice work coming out on Pseudomonas in human infections of the cystic fibrosis lung now. So I think trying to get the social microbe world to into more realistic settings is one of the most important things in the field. But the few cases that have been done so far, you basically get identical results in the lab.
Yeah. If you set up the experiment right, you get the same results. Okay, many questions here. There was a very eager hand in the back there. The guys who were waving, that's one, yeah.
Hi, my question I think is kind of related to her comment. I'm having problems understanding this idea, mostly in bacterial populations, of how clonality actually results in cooperation. The idea that I had was that the more equal you are, the more likely you are to compete for the same resources. That diversity somehow gives complementation and more social structure. I don't know, maybe I'm not understanding correctly what clonality means or something.
I think we're thinking slightly different things. So the kind of thing you're thinking about, to me, I would say that will influence the costs and benefits of cooperation. And, well, actually, I've got a slide that answers that. Go back far enough. God, too far back. So when I talked about this experiment here, I was comparing a scenario where you culture them under conditions of relatively low relatedness, relatively high relatedness. There was also two other treatments. What those other treatments were was the amount of local competition. So relatedness matters. You should favor cooperation towards higher relatives. But I'm sort of oversimplifying slightly. because in some cases that can be cancelled out by competition, okay? And if higher relatedness also leads to more competition between relatives, that can cancel out that effect. And that was that was the main point of this experiment. So here you've got high relatedness, low competition between relatives. Here you've got high relatedness, high competition between relatives. And there you've got the same two things with that. So I think these are multiple things that can happen at the same time, and you have to account for them. There was a sort of oversimplistic idea that just sort of limited dispersal should favor cooperation because it would get relatives together, but it can also increase competition. So you need certain types of population structures that will give you one but not too much of the other. Does that make sense?
Thank you. Do you know if during the formation of a multicellular organism exists a kind of cell lineage competition?
Well, the crucial thing I would say is that in the evolution of obligate multicellularity has only happened within clonal groups. So there isn't really selection for competition.
I'm talking
Or do you mean, but more
specific in embryogenesis.
Okay, but I was going to say, but then in the facultative multicellular cases, so for example something like the slime mode, there does seem to be examples of some cells being better competitions for others. So I'd say we would expect cell lineage competition in cases where you've got non-clonality, but not in the cases where you've got clonality.
In the black and gray striped sweater.
Yeah, thanks. You mentioned an example of some kind of punishment mechanism where the nodules of the plant that weren't fixating nitrogen anymore were like cutting their nutrient supply, and my question is if you know another type of mechanism in another organism where they punish like elements of the society that are not cooperating anymore, and how did these punish mechanisms arrive and how did it affect the establishment of societies?
Since that work on the legume and rhizobia, there's been a lot of studies arising showing the same sort of thing. So an example of something you get in Mexico is the interaction between figs and the wasps that pollinate them. And so there's a nice study been done on some Panamanian fig wasps actually showing that in particular figs where the wasps aren't doing enough pollinating, they're laying lots of eggs, but they're not pollinating. Do people know the biology of the fig wasp system? I guess there's lots of people that might not. So fig trees, basically loads of species of them produce fruit, wasps come into them, they pollinate the flowers within the fruit, which is essentially an enclosed flower, and then they also lay eggs. And so eggs develop, and so what could be seeds develop into either actual seeds or wasps. The figs want wasps to carry their pollen to other trees because they're monoecious, most of them, but also at the same time they want seeds. In cases where the wasps don't do enough pollination, they're not doing enough good for the fig, they're behaving a bit selfishly, the fig tree just drops those fruit. Another example would be, we've recently found that a sort of similar, well, slightly different things happen in mycorrhizae. So a nice thing about the legume rhizobia is you've got one plant interacting with lots of different rhizobia, it drops the less cooperative rhizobia. In mycorrhizae plant interactions, you've got multiple partners interacting with multiple partners in a network. And what seems to go on there is more like a sort of market. It's a bit of a stupid analogy, but a bit more like a human market. So plants preferentially give more carbon to fungi that are giving them more phosphorus, and the fungi are also doing the same in reverse. They're preferentially giving resources to more cooperative individuals. So I think the second part of your question, so where did that come from? So we can see how these things have evolved into quite nice mechanisms for maintaining cooperation. How they got there is a really interesting issue, I think, actually. And cuts to a lot of the sort of pre-adaptation. When did these things happen? For example, in rhizobia, we know now the legumes actively cut off the oxygen supply and stop supplying resources to less cooperative nodules. There's a very strong behavior going on. But that might have originally evolved much more gradually, just growing slightly less, just slightly less resources to less successful areas. You can see how those mechanisms could be built on kind of existing things that were there, maybe even before the symbiosis. But we don't know enough about that yet.
Okay, I'm going to take a couple more questions. You in the scarf, and then John.
How does the Castellium cell know that other cell is its clone? Because I can imagine that they express some proteins on the surface and recognize each other, but there must be other genes that are not expressed and that may be different between them.
My short and safe answer would be I don't know. My slightly longer answer is they have, Dave Queller and Joan Strassman's group have been looking into the details of that, and I think it's to do with cell surface genes. Yeah. And there's a couple of different, it's slightly complicated in that they've found different, very different recognition systems in very different species. Some of them it's very single gene specific, some it seems to be more general, acting on sort of multiple genes. But they've published some really nice work on that that's worth looking up.
Okay, I have maybe a fun but certainly provocative question. What are your thoughts about the evolution of social groups or sociality in humans with respect to monogamy?
That's a good question, and it's funny, when we published the bird promiscuity data, all the media ever asked us, what about humans? And so Charlie Cornwallis, the first author of that data, has started collecting data on different human populations and promiscuity and cooperation. I've no idea what the results are yet. Human social evolution is a lot more complicated, though. We have social learning, cultural evolution, which changes things. We haven't got the same sort of, I don't need to tell you this, we don't have the same genetic theory applying so clearly all the time. And so while we might expect similar things, there's also reasons it might not work.
So the hegemony of humans being monogamous ancestrally, it probably has some effect on that thinking. So it'll be fun to see those data. So
yeah.
Cool.
Okay, thank you everyone. It's time for lunch. Okay, there's a small announcement. If you could maybe please walk outside. We're going to take a panoramic picture for the event. So those of you who want to participate, please join us just right outside. Thank you.